the attendance is becoming thinner and thinner by day by day. I think you should call everyone. I think who is missing? Our own students. Otherwise, you don't get biryani today. Okay, so good morning. So this is a little different, mostly covered our research, our laboratory work. One of the student, Mr. Dr. Jayendar, who has done all this work. And I kept the photo, the work always I dedicate this work to my PhD supervisor, Prasanna Mohanty from JNU, Jawaharlal Nehru University, who is no more, but I know. So I'm trying to walk into his shoes, but I hope I did, but still a lot more to do. Okay, so here, how the chloroplast behaves, or how chloroplast getting exposed to any drought, the stress is called drought stress, and we used a model plant, the Pisum satim. You know that Pisum satim, chickpea also we call it as. And this is a little bit different. Here is, can they keep memory? So when the stress is, when the plants are, even in an animal system. So when you expose, let's take somebody is affected with cancer. So even if the treatment is there, still that, the side effects will be remaining. Similarly, on plants, most of the research is derived from the plants because, the mechanism is the same. So, but now I'm giving an example from, animal to plant, plant to animal. So now if you see the climate change that, I think, Professor Tripathi has explained the various climatic changes, the plants or animals, they get affected with the cycle. So now, what is the drought does? Drought means less water, less rains. So now the mostly affected, the first affected people are mostly farmers because they are the one who always in the farms and looking for most of the dry lands, they with rain-fed crops. If there is no rain, they'll be looking like that, waiting for the rain. And people are walking on the fields. These are I think rice fields, or maybe wheat field. So, and we can see all these cracks, and these are the can affect the land structure, the soil, the composition also it varies. If it is, that's most of the time in Hyderabad, there are many of the Indian scenario, when it's summer it comes, most of the plants die. Your garden, whatever garden you grow, all of them, having troubles. So if you look at the scenario of, another 20, 40, maybe, another 15 years, even India is going to affect with, dry land. You can see here the red marks. So all these red, and most of the countries, almost 54% of the agricultural lands is going to be affected by the drought. So now, what are the possible mechanisms in plants under drought stress? I mean, these are all known, that what happens if the drought is affected by the plants. So now, what the first one is, when the drought is affected or drought stress reduces the water use availability, because there is no water. You can see here water, and the signaling hormone ABA signaling will be increased. I mean, these one, these are all, and stomatal closure. The stomata get closer in order to protect our transmission, transpiration to, not to evaporate water. Diminished CO2 flux, that means CO2 efflux will be reduced, and all these photosynthetically get affected, and the ROS, reactive oxygen species, increases to protect the plant, and a lot of they see the attack on photosystems, and obstructed the synthesis of ATP. That means energy is will not synthesize. And if you go into the lower water potential will be, affect the plant. Lower ribosco activity. I mean, if ribosco activity is reduced, and obviously carbohydrate gets less carbohydrates, and leads to many other, other biomolecules also will not synthesize. And you can also see all these one, photosystem 1 and photosystem 2 decrease in water use efficiency. There is non-photochemical quenching, and some of the proteins, they will be overexpressed, non-photochemical quenching. And PSBS is again a stress-related protein, will be expressed to protect the plant. This is a protective system. And also there is a stress-regulating, the proteins or genes. The expression of LIA, genes and transporter genes will be, they express in order to protect the plants. Okay, so ultimately what it does, the less of, the photosynthesis will be reduced. So we have taken here is a P is a model. You all know that, all of us now the season now, it comes now P, you get nice P's. Now because there is a less research is, happening on, the P connecting to the drought stress. So, it contain, all of you know that, rich in protein, sugars, carbohydrates, and, we wanted to see, I mean, there are a lot of, nutritional value and a lot of medicinal values, one could consume, we all get all this one. So, I mean, these are the things I'm going to cover. And what Jayendra did here is, he induced natural drought stress. So not watering the plants for, after growing, we cannot, if you start with, if you don't add water, there won't be any seed germination. Once the seed germination has happened after 14 days, he has given 8 days a drought, then again, wanted to check whether it is going to be recover the plants. So then again 7 days will be, they watered again, re-watering. So then we test all this one, the photosynthesis, biochemical, molecular, omic studies. Entire, this project was, we have studied extensively, entire, we have addressed most of the, the biochemical, molecular, physiology, biophysics, all of them we have addressed. We have published a couple of papers in this topic. So, very interestingly here, now 8 days when you give plants, there is a plants get wilted, the leaves get wilted, and then after 7 days, so we keep one control. That means we don't induce any stress. We follow all regular watering. The other one we follow this one. The 7 day, why we have used 7 days? After 7 days, it gets flower. So we cannot continue. That is why. That's why we cannot use. But the recovery rate is not much. Okay. Anyway, so now what we did here, I'm not going to. This you all know that. I think this is the. You have measured already this one. It tells about the photochemistry of photosystem 2. There is a OJ IP curves. Each component will tell about the photosynthetic water oxidation and the PQ pool reduction and sending these 1 electrons to photosystem 1. So the data what it suggests here, this Fe bar Fm is a total photochemical reaction of photosystem 2, where you can see the day 0, this is the induction of the drought, day 2, day 4, day 6, you see that not much change. So when the day 8, I mean that after 2 days, there is a significant change in photochemical. This change is very significant, 0.85 to something like 6.1, about 1 difference, 0.1 difference is a significant difference. Then after watering, re-watering, it is reaching but not up to the control level. So then these are all talk about these two water oxidation complexes. Obviously, water oxidation is nothing but a splitting of water. So when there is no water, obviously the water splitting process get affected, where this increase is nothing but when you see here 100%. Now we use, these are the internal control, any in the protein chemistry. So if you don't have histone, histone you can use it, but still we use like 50, 100%. Let's say if you load 1 micromolar, 1 microliter or 1 microgram, and you use 0.5, then you use 0.25 grams, whatever milligrams or micrograms. So that is the dilution. It's called dilution. When you see here drought, there is a PSBO, one of the subunit, and PSBP, one of the subunit. There is about 50% has been decreased, which is an indication that the water oxidation complex is getting affected. Not only that one, we also looking at this one, we can tell many other things. There is a disturbance or disturbance between the pigment to pigment and pigment to protein, that we will discuss a little bit later. So similarly, whatever the handy P data, we can also, there is a PAM instrument, it also tells about electron transport. This is the electron transport and yield of photosystem 2. So this tells about that there is a decrease, significant decrease at the day 8. You can see very significant decrease is there. And similarly, the non-photochemical quenching is nothing but a excess energy is quenched. The excess energy, that is very important. Plants get protected. When you, let's say, all of you, if it is too much summer, okay, especially the average of Indian summer will be about 35 to 40 degrees. So you cannot stay in the summer, 10, 15 minutes. Can you stay in 15 minutes in summer light? You cannot. What you do, you go to shade. But plant doesn't have that mechanism. They cannot go to shade. They cannot walk. So what they do, that anything light is falling on the leaf, they have a mechanism they have developed that is called non-photochemical quenching. That excess energy, whatever, I take only this much water. Let's take a water or whatever. After that, I reject. Right? When you feel your stomach is feeling hungry, you take only the amount which you require. Even if you keep a food, bowl, big bowls, or we have mandi. Today is going to get mandi. So if you put mandi in front of you, you cannot eat entire mandi. So you eat only the 10% or to 15%. But the same thing, what it does here, the cyclic, the non-photochemical quenching is around here actually. So this place, and these are the two very important subunits, that PSBS play major role in increasing the the delta pH, that is called hydrogen ion protons. The protons get increased, and this balances the cyclic electron transport. This switch now from linear to cyclic electron transport during the non-photochemical quenching. Is it clear for you? Okay. Okay. Then maybe photosystem 1 also getting affected. I don't want to go into more. So what it does, So why they are doing it? So the question is why, there is change in photosystem, why? Mainly, you all know that the main culprit is ROS, reactive oxygen species. So what it does, there are, these are the reactive oxygen species generated by the plants, singlet toxin, peroxides, and hydroxyl radicals and H2O2. So these radicals are, mostly affected the plant, the plants get affected if they generate all these one. This is called oxidative stress signals, all these signals. So whether all of them are producing by the, the drought stress, but that's what, we'll see here. So what we have done all these superoxides, H2O2, this APX, catalase, and SOD. You can see here a significant increase, many folds of increase of all these is an indication that these are all playing a major role in protecting, against, the drought, but, the enzymes are there to scavenge the reactive oxygen species, but still, plants are able to survive. That's what I said. Plants are able to survive. One is non-photochemical quenching. There is other one reactive oxygen. There are many other things we'll discuss. Okay, so now the same thing we showed using Western blotting. Western blotting is nothing but antibodies. We against these proteins, we have antibodies. So we tested, the significant increase of catalase, manganese SOD, APX, and copper zinc SOD. You can see this in 100% drought. They have a significant increase. We showed this one in the relative increase in the graph. So what it does, this one, we can read this one. There is a decrease in photosynthesis, and because the increase of ROS and non-photochemical quenching. That is the summary of this one. So now, so I said oxidative stress appears to be reason for induced disassembly and aggregation of photosystems. So the proteins get aggregated. That's what we're going to discuss now. So this is, I think you are done yesterday, this experiment? Already loaded? Okay, we are going to load today. So now just let me know, they might have explained Pavitra and other one. So the anywhere in a chlamydomonas or in algal system or the cyanobacteria or the higher plants, they have this, Professor Tripathi has explained very well, stroma lamella, grana core, granum margens, all of them. So in which inside in thylakoids you have a complexes like photosystem 1, photosystem 2, light arresting complexes, photosystem 2 dimers, photosystem 2 monomers, and LHC2 trimers and LHC2 monomers, and ATP synthase complexes. All these can be separated using blue native gel electrophoresis, which Pavitra is explained yesterday, right? Pavitra, you have explained? Explain is not done. Okay, now this is another one, you can see here. So these are the complexes we could able to separate from the blue native gel. We isolate the thylakoids. Thylakoids, right, chloroplast. You disturb the chloroplast, you get thylakoids with using differential centrifugation. After that, we use, they're going to explain about, the beta-DM, the detergent, which the detergents will be one. It kind of, it makes a micelles inside micelles. The micelles will create, loosening of the complexes, and through electrophoretic mobility, you can separate all these complexes. These are blue native gel, similar to SDS phase. It's also called a native pair. So you don't add here, denaturing agent, beta-mercaptoethanol. Without that, so you can separate these complexes. So all these complexes, PS1 NDH complex, PS2 LH2 super complex, big complexes, PS1 dimers, I said, ATP synthase, PS monomers, cytochrome B6F complex. Whatever you see the thylakoid, the composition, schematic representation, which I showed you last class, all this can be separated using this one. You see, I told you trimer, dimer, monomers. So, that's what we are going to see. What happens to these complexes under drought stress, which you can see later, in the next slide. So, this is the one. So, drought, control, and recovery. The thylakoids, are separated using beta-DM, but electrophoretic mobility, all these complexes. So, I mean, drought also, most of the complexes are almost intact, but there are some, differences, especially the dimer, LS2 trimer, dimer is little increased. I mean, this is only Comesi strain, both are same. To see much clarity, we have done the Comesi. And there are some, especially the super complexes. The complexes, they have super complex means bigger complex, larger complexes got affected by the drought. So, that is that because of that, you see whatever the change in the photosynthetic activity. So, because change in these, they become looser, right? So, like a spring, like you do it like this. If you keep like that, that means they will be stretching out. That stretching is the one you could see here. When it's stretching, this is a spring structure. I mean, it comes back later. Once you put water, it comes back to the regular natural conditions. You can see this one. So, that is what we could see. The drought is inducing the change in super complexes, this one. So, then further what we did, I mean, there is also, you can see that one, the trimeric complex also little bit increased compared to control. And when coming back, they change these complexes, which you can also see here. So, I said, aggregation. The proteins are getting aggregated. So, now what to show that one? I mean, we have done all these very interesting, to understand the aggregation. We solubilize the thylakoids or anything. You can apply this one to any research, not only the chloroplast, even in many other studies. So, like, this can be like any disease, even human diseases also. So, you just, if there is anything membrane related complexes. So, if you want to understand what are the markers to markers against specific disease, to probe that one, you have to go with, the proteins, otherwise, you go with proteomic data or whatever. So, what we did, these solubilized fractions, again we run the SDS page, then probe with the antibody, and where you can see here, these are some of the PSA, B, and and also the CP43, 47, LHC-B2, LHC-B5, and all these blue color has enhanced into drought conditions. When you see this one, 100%, 100%, you can see here, they are significantly increased. That means when the solubulated fraction and there's a precipitate in the down, when you get centrifuge in the down, in the soluble one and down, you will have a precipitated. That precipitated is nothing but aggregations. Proteins get aggregated, tend to aggregate if there is any kind of stress. It can be applied to plant or animal or any kind of these human samples also. So, these proteins are playing a major role in aggregation form. Why they have aggregated, that is again a story that because of ROS, they are getting aggregated. So, we did use that one, using confocal microscopy, where, you can see this is the control and this is the one, the treated, that means drought. You can see the clumps, very big clumps. I mean, the different magnification, you can see a very, red spots where you cannot see much, but this is an indication. But this again we have run the SDS page to see this one. What are the proteins are getting, aggregated. So, what we said, these are the proteins are getting aggregated. So, hence, we showed other, the green gels, and where, in the reverse recovery, you don't see much in this one. That means, and it comes back to the plants are always, I say that they are a beautiful mechanism. Like not an animal, like, plants, even if the leaves are falling, pour water, again, regenerate and comes back to natural environment conditions. But not always, but if it is a disease like pathological effects, very difficult. But abiotic stress, they can come back to original conditions. Okay, so we showed again we using a circular dichroism. We are not measured it, may not yet. Today, no? Okay. So this is the one, you can say that pigment protein complexes can be understand pigment interactions, and pigment protein interaction also can be understand by using circular dichroism spectroscopy, where we showed a lot of significant change in the pigment composition. Pigment, you see that in the drought it significantly affected this region, that is a mostly chlorophyll A region. And similarly, there is a carotenoids also getting affected. This is the carotenoid region, this is also getting affected. So there is a change in chlorophenol and change in chlorophylls. So this is again, I think Professor Tripathi was talking about these the structures. This electron microscope structure, you can see nice. Now this is the control, and this is the drought, and this one is the recovery. So where let's take this one is the same magnification, and this one same magnification, this one same magnification. This is same magnification, and same magnification. So and you can see here this size, okay, this size and this size has significantly increased. That means it's kind of a swollen. And also you say that you can also become, like because of there is a pigment interaction has been changed, and pigment protein also has been changed. So that's why it is becoming loosening. The stacking is whatever they said yesterday was nicely explained to you, right? So there is becoming because electrostatic interaction is there is change in electrostatic interaction. They become loosened because the ion mobility has changed. So that's why, because the drought, there's more of water is going out, so there is a change in the mobility of ions. So there you can see the lot of change in this one. It's increased, and whereas in reversible, this comes back to the native state. So similarly, what we did, just created a model. This is the model where you see that GIAN stacks the repeated distance. So this is the one I said increased. Those repeated distances you can see here, it is increased in the drought stress. That is again I said it is because of less of water and many other things, many factors that we have showed. Maybe now you can share the paper, you can going through that. Okay, I'm not going to discuss this one because already Ichiro Takahashi is explained, and now we wanted to see what is happening to the photosystem 1. So on photosystem 1 and photosystem 2, the structures, these are the structural the components, the dimensions, and these are the photosystem 2 structure compounds and their proteins, and we showed all of them using Western blotting. We probed all this one, and you can see their change when it is expected to drought, and how they have changed. You can see here some of the proteins like SCP43, and also the PSBO, PSBB already showed you, and there are also like like the LHC2B, that means photosystem 2 light harvesting complex also you can see here significantly changed, and maybe LHC5 also changed, LHC6 also changed, and as I said, dehydrin and aquaporins are the markers for drought. Reviewers will ask what you have used, what now you have induced drought. What is the evidence? How do you prove that this is affected to the drought? So to show that one, the aquaporins and dehydrin is the one you see here, a very significant increase. There is no dehydrin is nothing but hydration. Dehydration. So when there is no water, these proteins get overexpressed, and you can see here very significant increase in this subunit. And similarly, aquaporins also will be have increased here. And now similarly, photosystem 1 There are some proteins, PSA and some little bit PSB and some of the proteins like PSAF and some they have reduced. You can see especially these two PSA B and the F they changed, and mostly LHC1 is almost like LHCA2 is reduced significantly, and LHCA4 is little bit reduced, but otherwise all of them are stable. These are the light arresting complex of photosystem 1, they change. So, okay, to conclude to this objective here, you can see here in all these biochemical, biophysical, the NPQ is induced, and these some of the specific proteins are getting overexpressed or during aggregation process, and these I think Lea dehydrogen proteins are expressed, and some of the light arresting photosystem 2, photosystem 1 proteins core get aggregated and drought says. So, yeah. So now this you can check this paper. If you want to understand more, we have given all the details in this paper. Okay, now this Echiro Takahashi is explaining about the complexes. What is happening inside the complexes? We have separated them using sucrose density gradient. You can see these complexes, F1, F2, F3, F4 is there are fractions. So F1 is light arresting complex 2, F3 is PS2, F2 in the F3, you can see here F1 LHC2, F2 PS2 core dimer, and F3 PS1 LHC complexes, and last one is F4 is PS1 LHC1 complexes. So I don't want to go into this one. I mean, I will skip this one. Only what I want to tell you here, the pigments. There are, and I mean just to conclude this one, I don't want to go into more onto this one. So what from this sucrose density gradient, what we said here is there are different. I mean, you have seen the fractions: LHC2, PS2 core dimer, and PS1 and PS2 and light arresting complexes, and last one is PS1 LH1 complexes. And these complexes, they express, I mean, different carotenoid pigments and chlorophyll pigments. And there are differential change in the pigments, which leads the change in the protein and pigment complexes. Change in the pigment. That's what we wanted to say. I mean, if you want to go through it, we can go through our laboratory work and lot of published. So now that, and we also checked, using RT-PCR or Western blotting, reconfirm whether whatever we have done is correct. So we used ZEP means zeaxanthin, viloxanthin, all this one you can see in the drought condition, they have significantly increased, which is we can say that a marker for identification of non-photochemical quenching, and also the increase in the carotenoid biosynthesis during drought conditions. And the same thing, we have tested all the genes by using the RT-PCR, where you can see that most of these pigments, that means let's take a beta carotene to zeaxanthin, you can see the CHYB, you can see the increased here, and and if you can see this in some of the genes, xanthophyll cycle, and the CHYB, you can see here is increased, and PDS is mostly the conversion from this one, phytin to lycopene itself is increased significantly, which is an indication that the whatever the pigments coming from the chlorophylls are converting to carotenoids, and these are the one, all these compounds have increased. Okay, so now I mean this work was, we can also share this paper, the previous one we published in Plant Cell Physiology, and this work is now, it's also in the web, recently published another good paper, 3 good papers. It's a plant cell environment. There is another paper. We showed all this one, what is the connection between lipids, membrane lipids, how the membrane lipids are playing a role in photoprotection against drought stress. So, if you look at this one, possible mechanism plants under drought stress and polar and membrane lipids, you all know that membrane lipids, we have 4 membrane lipids, MGDG, DGDG, SQDG and PG. So, these are 4 major membrane lipids. So, when the plants are affected to drought, so there will be a production of ROS, all this we discussed. They can affect the DNA, protein, lipids, and then the lipids, they will change the lipids like MGDG, DGDG, SQDG, PG, and there is PI. Now there is some plants. So what they do, always the lipids, when they convert into different forms. Why they convert? Any of you can tell? Any lipids like a carbohydrates also they convert into one form to another form. These are metabolic conversions. So this is mainly because to produce ATP, the energy, or this is a protection mechanism. So that if the lipids are converted to triacyl glycerol, there will be a lot of energy generated. This energy, or this also what maybe this, because directly the ROS is getting affected to the lipids. So now another form they trying to accumulate. So this is the accumulation is triacyl glycerol. They accumulate, and when plant is required, let's say a plant is getting stressed, let's say too much stress, the lot of glycerol is accumulated. After the accumulation, what it does, when the plant you water it again, these guys can convert into again lipids to the MGD. Similarly, this is one way to the lipid getting affected. Then another one, attack on photosynthetic membranes, where you could have seen that first 2 objectives which I told you that all is getting affected. So these are all the antioxidant protectants. There are osmoprotectants. There are antioxidant mechanism. They're all the stress defense mechanism, SOD, APX, which I showed in the first chapter. The objective proline, they will be all involved. They increase during the stress condition to protect all the plants or animal, even an animal system also. The same kind. The mechanism is same in animal system. Okay, so what we did here, just want to show that, in the drought conditions, this is biomass, we already, less biomass, and carbohydrate content is increased, is significant. It has to increase because there is a, the alternatively now it accumulates more carbohydrates, and starch content, since carbohydrates, starch also increases, and the sugars, these we have identified sugars, and proline, you see that a high amount of proline, I don't know how many folds, 5 to 30 means 6, 5, 6, 7 folds more, and leaf area reduced. Obviously, leaf area when it's getting affected is reduced. So we also tested other, they have increased significantly. So then what we did, we separated the membrane complexes or membrane lipids using TLC, thin layer chromatography. So after separation, we have separated all these the lipids, MGDG, DGDG, SQDG, and TGDS, trimethyl homoserine. Sometimes we get, sometimes depending on the plant, and phosphatidyl glycerol. So they looks like that. Then you can see here. First thing what we did, the triacyl glycerol separation. One is the triacyl. This is basically organic solvent. Different organic solvents matters. So you can see here in drought stress, MGDG is reduced when you see this one, and DGDG and PG, they all that means they have converted. They have degraded actually. So this degraded, and this is coming back to the control level, but not up to the one in the recovery. So and SQDG also changed. So what we can see here, when you see these in TAG, that's what we wanted to show here. And they have increased. This is the these two are the TAG, that means triacylglycerols. In control, not much, they have increased slightly. This is indication that they are converting into these, they convert into TAG. Maybe not all of them, but some part. Okay, now we did this one to 2D, I mean the one dimension, second dimension TLC, where you can see here, I mean the drought, MGDG, there is a change, the DGDG significantly changed, and the SQDG, you can see that very, almost more than 60% has changed, and it is reversing in recovery conditions. So we did all this one fatty acid analysis and showed, you can see here, some of the control, recovery, for 42% you can, drought is significantly increased the fatty acid composition. That again, when the fatty acid is degrading, that means beta oxidation happens, there will be a lot of ATP molecules, the energy generated, that energy is utilized by the plant to cope up to the plant, to the stress. And you can see that significant increase in saturated fatty acids. So then this is the outcome of this one. The conversion of these, all these one, I said acetyl-CoA is the one. Through acetyl-CoA, you get all these, the final products. So these PGAT and DGAT, these are the one. The conversion of membrane complexes comes somewhere, here. They convert into acetyl-CoA, then it comes here. And here, and from there, before the generating a triacylglycerol, the PGAT and DGAT are the one playing major role to involve in conversion of the TAG. Where we wanted to show here, you can see these 100% here, compared to here, almost more than twofold has increased. This one and this one. Similarly, you can see here this one and this one. And also we showed something, ATG is also increased, autophagy is, that is a different story, and I am not going to discuss. Autophagy also is significantly increased in drought stress. So the meaning of this one here is the lipids, membrane complexes are, I mean not only membrane, any other lipids also is converted to TAG, apart from membrane complexes. This one, the one we have seen, polyunsaturated fatty acid. Polyunsaturated fatty acids are significantly increased. These polyunsaturated fatty acids are involved in conversion of TAG, where they are coming, where polyunsaturated lipids are coming, they are coming from membrane complex, membrane lipids. And other, DAG also is increased because maybe some other lipids also coming. Not only the membrane, maybe other lipids also converting into TAG. Okay. To conclude this one, these are the, the conclusions. The TAG accumulation again coming from membrane complexes. There are a lot of significantly increased the osmolytes like proline and even a lot of glycerols they have accumulated. Okay, so now little bit about omic studies. I mean too much of you have I will not talk about all these methodology. We did a lot of omics, like all omics, genomics, transcriptome, proteome, metabolome. We connected all of them and understand and to understand the markers, and these markers now will be using to develop the against drought. Now we'll be using we'll be using that one in many, even chlamydomonas, you can use those markers to develop, stress resistant plants or stress resistant algal system. So I will not go into deep into all these analysis, I will just only go with some of the markers where we have, these are the, upregulation of transcription factors. What we have seen, there are the, ABA is one of the major one, and some of the heat shock proteins, and we will also see that these are the drought, you see that control and drought treated, where you can see these genes, PSB and PSBS, which I in Western blot also we showed some of the the markers, they have increased significantly, and now these are again the markers, they are significantly increased. We want, And some of the photosynthetic pigment, these are very, you cannot read them. Some of the photosynthetic genes also been significantly downregulated and significantly upregulated some few of the photosynthetic proteins. Then we did photosynthetic genes or transcripts. So now we did the proteomic analysis using the ITRAC method, where the samples was analyzed by UC Berkeley. We sent the samples to US. They are given the data. So where I will not discuss all of them, because to show to the genes and what are the genes, the same genes we have analyzed with the proteins, and you can see all these one is similar to the gene expression or transcriptal expression. The proteins also getting similarly getting overexpressed in some of the places, and you can also similarly we tested with the transcriptome and the analyzed together. Some of the, see the photosynthetic protein, these are all photosynthetic proteins, PSAB, that means photosystem 1, photosystem 1D, photosystem 1E, photosystem 1F, they are all significantly decreased, which is agreement with the other biochemical data. Okay, this, to show this one, this I already showed, but to show again, this is the Western blotting data, which is matching with the proteomic data. So then again, we tested many other, these are all the, again, probably you might be knowing all these one, DNAJ, thyroidexin, thyroidexin, COX, NOD, nitric oxide related genes. They play major role in stress regulated mechanisms. So what we are now working and trying to understand, these genes, can we improve the plants, or can we improve the algal system against a stress condition? But they are very specific to drought. There are also some other genes. This is nothing to do with protein. There are for the photosynthesis. There are some other. They can also see here some of the genes of the thyroidexin, iron sulfur cluster related cytochrome 450. They are also increased significantly protein side. So we did a lot of network analysis, which I'm, network analysis will tell about whether whatever the proteins are, the genes, how much interaction is there from one protein to another protein, which is, you guys maybe know much better than me. Okay, we did all the metabolomic analysis. We have identified all these one. I'm not telling that we have identified 35 metabolites and 3 groups. All these groups were significantly increased in drought stress. You can see this in carbohydrates, polyols, and other metabolites. So you can play around these metabolic, and improve, or let's say they have increased all these one. Maybe you can, the genes of these specific metabolites, you can also manipulate. That means you can knock out, or you can see that mostly do that knockout, and the biggest is more overexpressed, maybe silence it, then see that if you can able to protect, or since I don't know, there was yesterday a beautiful talk was given by somebody from US scientist. So the CRISPR is the one now telling multi-genes you can play around. So multi-genes, about 10 genes at a time, you can you can silence, or you can knock out. So let's say these are one specific genes, you silence it and see that if they can able to protect against stress. Maybe in future we'll be doing all this one. Okay, so to conclude all this one, you can read this here. Our transcript data revealed that most of the photosynthetic genes are downregulated, and stress markers genes, especially dehydrin, PSBS, and Shisha proteins, and aquaporins, and are these are increased, and majorly photosynthetic genes are downregulated, because when you can see that photosynthesis getting affected, so where we could see all the most of the proteins are related to photosynthesis, they decrease. And the ABA responsive elements, ethylene responsive transcript factor or upregulated in droughts, that has to be there. If the ABA, the play hormonal effect is very important. And the same thing, proteins, both photohistone 1 and photohistone 2, the proteins getting affected, and ROS and the heat shock proteins enhance performance, leaf drought conditions, and some other proteins are also, they're kind of stable or increased. So all this one, the carbohydrate polyols, so they have increased to maintain the cell integrity. So that's why always the these are osmolytes. You can also say the osmotic. They maintain the osmotic because of these, the carbohydrates and polyol. If they are not increased, and the plants die immediately. When you give a stress, they will get immediately affected. So overall, this is the summary of the entire study of the drought stress on chloroplast, how the chloroplast getting affected, chloroplast metabolites, proteome, the transcriptome, other physiology and biochemistry, biophysics getting affected. You can clearly see that one. See that downwards is decreased and upwards are increased. You can take picture, and now we can also, anyway, we will be sharing all this material with you. So with this, I conclude, and if you have any questions, I'll be happy to answer. Any questions? I said these studies can be implemented not only plants and animals, especially the ROS mechanism and their protein aggregation, and the metabolome studies can be applicable to even humans or animal studies also. So likewise, only the discovery happens animal to plant. Many noble laureates, the autophagy Nobel has come from plant to animal. So they are tested, they have taken plant genes. Then the Japanese guy got Nobel Prize. The many other a lot of Nobel laureates are all getting discoveries, reading more articles from plant side. At the same time, sometimes it also happens from animal to plant also. The recent studies like movement, plants also can speak, right? Have you studied that article? They communicate. So they also cry. Plants cry. Now that's what they prove that the studies are proving that a couple of nature papers has come last 2 years back. Yes. Yeah, we use the markers. Basically markers, known markers. Yeah, markers we have to give. That's what we use TAG, triacylglycerol marker. Otherwise, it's difficult. So then MGDG, DGDG, SQDG, and all you have to use markers. Markers, the literature. I mean, there is already a standard protocol. If it is MGDG, DGDG, SQDG, these, it is known. You know that it's a separation. Based on the literature, you can identify. But we had a lot of difficulties publishing this one. They were keep asking us to show. I mean, we believe, but even if you cite literature, they say that how can we believe that your whatever the bands are, when we have to do it. Actually, we didn't show here, but in the publication we showed using standards. Otherwise, when you submit to big journals, it's difficult. Now, especially these lipids, when you are running on TLC, you have to use standards. Without standards, now even if going to LCMS, it gives different. It gives only fatty acid, right? It won't give you lipids. Mostly fatty acid, right? So, but there is one more you can use it, FTIR. FTIR. But FTR is only functional group, yes. Now you cannot get FTR also, but the lipids based on that, there also it is qualitative only. Quantitative, I mean, um, to really identify, you need to use without markers, difficult. But many people, they agree, with the TLC. What when you run TLC, it's already known. MGDG separates like that. People know that, but like a crazy like me, I may, like if I'm reviewing, I want to reject the paper, I will ask, okay, you have to do it. So those are wise men, wise people, yes, okay, it's a known procedure, why to, bother them. So it's like that. But better to have the markers. My students also can ask questions. So no questions. Okay, then we'll break for tea. So next Professor Tripathi will come. 12 o'clock, Echiro Takashi will come.
Are you enjoying lectures or not? Good. Huh? Enjoying, that's very important. Okay. Yeah, we are talking other day that cyanobacteria-like organisms became a chloroplast in the process of endosymbiosis, right? And for billions of years, became a chloroplast. So as I told you, once a living organism like cyanobacteria, which becomes endosymbiont, lives in a host cytoplasm, it doesn't need many genes anymore, right? Because it is living on a host cytoplasm. So he's provided all facilities, right? So therefore, it lost many genes completely. So you can compare cyanobacterial DNA and chloroplast DNA, much, much smaller than the cyanobacterial DNA. Okay, it's very, very small in size. We lost many genes. Okay, but then also not only completely lost, nucleus also wanted to have a central command over the chloroplast. Okay, and our same goes to mitochondria too. Okay, so then what happened? Then many genes were transferred from the nucleus, from the ancestor genome, that is cyanobacterial genome, to the nuclear genome. Okay, so the protein that are coded by the chloroplasts before, by the cyanobacteria before, is now being coded by the nucleus. Okay, and once it's coded by the nucleus, the messenger RNA is produced in the nucleus, then it comes out to through nuclear pores to the cytoplasm, where it translated on ATS ribosomes, and then post-translation they are imported into the chloroplast. Okay. And still there are some proteins that are coded by the chloroplast genome also. We are talking about other day, right? RBCL is the small subunit is coded by the nuclear genome, and large subunit coded by the chloroplast genome, okay, Rubisco. Therefore, Rubisco has been Rubisco small subunit because the major protein being transported, because 50% lipid, I told you, and 8 small units and 8 large subunits form one enzyme called Rubisco. Okay, so to build one enzyme Rubisco, with 50% lipid protein, chloroplast protein. Okay, Rubisco is the most abundant protein that is transported from the cytoplasm to the chloroplast. Okay, therefore, most of the model studies they look at it, look at Rubisco as a standard. Okay, from standard protein. Okay, for its translocation mechanism. Okay, all right, understood. There are many other genes we are not talking. I will talk about some, but I may not talk about it today also, depending on time. Okay, I don't have only 1 hour today. So there are also many other genes who are not only transported to the cytoplasm, they are also transported to the thalacid membrane, like light harvesting protein complexes, LFCs, right? They are transported to the thalacid membranes, and some are transported to thalacid lumen, right? Thalacid lumen, okay? So oxygen-evolving complex proteins, right? 33, 23, 18 kD proteins. They are all coded by nuclear genome. Post-translational have imported into the lumen of the thalacid. So it has to cross the outer membrane, intermembrane space, inner membrane, there is stroma, then it crosses the thalacid membrane, then it is the luminothalacid. Okay? But think out, there are many, many barriers for it to reach into the luminothalacid. So therefore, nature has evolved different kinds of mechanism, okay, to import different kinds of proteins, okay, into the chloroplast. But the topic has been given to me, general membrane, right?
Sir, I have a doubt.
Yes.
You told that the genes have been transported from the chloroplast to the nucleus, and then
No protein grows out from the chloroplast to the
You
nucleus.
said from the time of cyanobacteria, some of the genes are being produced by the nucleus now.
Yes.
Compared to the chloroplast. So what is the advantage of it? Because importing it back to the chloroplast is expensive process.
The advantages is this, that it has Mendelian inherited now, Mendelian fashion. If you remain in chloroplast, it will maternally inherited. Okay, that is one of the reasons why nature selected to put all the genes back into the wherever possible, back into nuclear genome for Mendelian inheritance. Okay, that is very important. Mendelian inheritance is very important component of plant breeding. Okay, cross breeding, cross pollination, right? We have come out. If you look at it, when in the process, nature has favored cross pollination. Okay, went out of the self pollination to cross pollination. Cross pollination happened among two different same plant, two different plants later on, right? And not only that, then plant became dioecious. Male plant, you see papaya, there are male papaya and female papaya plants. You know it? Male palms and female palms. You know it? So it has moved slowly from hermaphrodite condition, right, to the dioecious conditions. Okay, all right. So same thing also in animal evolution. Okay, we started with annelids, right, which is where mostly self-fertilization, annelids, right. If you come to that, still there are strains of but very lower organisms, we have self-fertilization in animal system. We have come out of that, and always there is cross-fertilization. Okay, you have men and women. Okay, there are no. Okay, so that has happened. Nature has favored this cross-fertilization to segregate gene pools, right, and then remix gene pools. Okay, so that evolution can go on. Okay, so this cross-pollination favored evolution. Okay, all right. Okay, so when let's say there are two kinds of protein import. One is co-translational protein import. Another is post-translational protein import. What is co-translational? As and when protein synthesized on the ribosome surface, okay, right? The proteins, it comes a signal sequence. They recognize endoplasmic reticulum, docks to endoplasmic reticulum directly, while it is still being synthesized in the ATS ribosomes. If you see most of the protein synthesized, all the messenger RNA or polyribosomes are bound to the endoplasmic reticulum. So as and when it's synthesized, the signal sequence of the protein docks to the endoplasmic reticulum or binds to the endoplasmic reticulum, then goes into the endoplasmic reticulum for post-translational processing, methylation, whatever takes place, the protein post-translational modification, alkylations, etc., all takes place in the endoplasmic reticulum. Then it comes out of endoplasmic reticulum, okay, whenever required, and it depends upon destination, okay. So remember there is no problem, okay, because synthesized and transported. You see, protein has to, if it's synthesized in the cytoplasm completely, before it attaches endoplasmic reticulum, okay, then it must attain its three-dimensional configuration, right? It is a cytoplasmic aquatic environment, right? Not hydrophobic environment, but aquatic environment. So protein folding will take place, no matter what, okay, in response to the cytoplasm. aquatic environment of the cytoplasm. Okay. So protein folding has to take place. And then protein has to partially unfold to go into the chloroplast. So there are many things we'll talk about it. So I'm not talking about co-transference protein import, if, mechanism and all, okay. That a completely different mechanism for the transport of proteins in the endoplasmic reticulum, okay. Okay, if you see, as I told you, polyribosomes are attached to the where? Endoplasmic reticulum, okay. But if you see chloroplast or mitochondria, no polyribosomes attached, polyribosomes are not attached to neither chloroplast nor mitochondria, okay. All right. So protein synthesis is taking place in the cytoplasm first, and then they are being imported into the chloroplast or mitochondria, okay. So both of them have therefore post-translational protein import, okay. All right, understandable? Okay, this is, I always, so this first time it's an endosymbiotic origin, right? Chloroplast DNA is a plastome, okay, contains 300 proteins, 100 proteins encoded by chloroplast DNA. Out of 3,000 proteins, 100 proteins encoded by chloroplast DNA. So minuscule amount, right? So 2,900 proteins are coded by the nuclear genome and post-translation reported into the chloroplast. Got it? Okay. I'll skip the slides. All you know about light regulation of chloroplast. I think Dr. Takahashi will talk about light regulation of chloroplast and all. So I'm not touching this point, okay? Okay. So this is the protein import apparatus present in the chloroplast membrane. You see, we are simply talking about protein that is going out from the cytoplasm here to the stroma here. This is thylakoid membrane. Outer membrane, bilayer outer membrane, bilayer inner membrane, okay, and then the stroma, okay. So the protein has to synthesize in cytoplasm, post-translationally has to be imported into through the outer membrane, through the intermembrane space, to the inner membrane, again to the cytoplasm, stroma, okay. So what it has? If you see protein, all the proteins are synthesized with the transit peptide. Okay, you see, red color is transit peptide. Just like signal sequence for the endoplasmic reticulum, here you have called transit peptide. Okay, this transit peptide recognizes where it has to go. Okay, and receptors present in the thylakoid membrane, okay, or in the envelope membrane. Here is envelope membrane, okay, envelope membrane chloroplast decides which protein to accept. Okay, so mutual selection of the transit peptide and the chloroplast. You see, both chloroplast protein and mitochondrial protein are synthesized in the cytoplasm, but only chloroplastic protein goes to chloroplast and mitochondrial protein goes to mitochondria. Cytochrome C does not go to chloroplast, nor rubisco small subunit goes to mitochondria. Not it. So there is selection process. Why is it so? We'll talk about it later. Okay. All right. So this is a transit peptide, and this is a protein. Okay, this protein is attached to HSP90 and HSP70. What is HSP? Heat suck proteins. They work like molecular chaperones. I talked about chaperones a few minutes ago, last class. This is the purpose I talk about is this, chaperones, okay. we need chaperones for providing proper conformation to the protein. Okay. So here, these two chaperones, okay, all right. Can you see here? If you cannot come to here, come here. Come here. Why you are sitting backwards? Those who are sitting back, come up. Here, come here. There is enough space. Come here. Yeah. Okay. You can, if you want, you can come also. Okay. So HSP 70 and HSP 90. These are the molecular chaperones. Okay. And these molecular chaperones keep the protein in transport competent form. Okay. If the protein completely folds and folds anomalously, okay, without molecular chaperones, it becomes transport incompetent. If after it's synthesized, okay, in ATS ribosomes, okay, attains 3-dimensional configuration without molecular chaperones, then it is transport incompetent. It cannot be transported into the chloroplast. And therefore, these are degraded. Any protein that is not does not bind to the appropriate molecular chaperones, they are transport incompetent, and they are ultimately degraded by proteasome system. Okay, they are ubiquitinated and goes to, and then they go to proteasome complex where they are degraded to individual amino acids. Okay, all right. We'll talk about it later on. So now you can see this import apparatus. What is their import apparatus? It is the term TUC. TUC stands for translocon in the outer chloroplast envelope membrane. TUC. Translocon in the outer chloroplast outer envelope membrane. TUC. TIC stands for translocon at the inner chloroplast envelope membrane. Okay? So TUC and TIC. TUC stands for outer membrane, TIC stands for inner membrane. So if say TIC 20, you immediately understand it present in the inner membrane of chloroplast. If say TUC 75 or TUC 34 or TUC 159, they are present in the outer envelope membrane of the chloroplast. Okay, all right. Same goes to mitochondria. Instead of mitochondria, you get TOM and TIM. Okay, TOM is the chloroplast outer membrane of mitochondria. TIM is the inner membrane of mitochondria. Okay, okay. So TUC 159, TUC 34. Has anybody taught you about anything about this import apparatus in this class, in this lecture series? Huh? Okay. All right. Okay, doesn't matter. Okay. So then what happens? Well, let me describe the process before you go. Okay, details. So what happens? First, the transit peptide comes and binds to the TUC 159. TUC 159 through TUC 34. So both TUC 34 and TUC 159 work as receptors, okay, to recognize the transit peptide. Okay, only if it is chloroplastic transit peptide, okay, they will bind with it. If it is mitochondrial transit peptide, they will not bind to it. Okay, so these receptors have the ability to discriminate chloroplast transit peptide and mitochondrial transit peptide. Understandable? Okay? All right. So once it TUC 159, then it goes inside. We'll talk not I'm describing here now. The TUC 75, which is barrel kind of protein, TUC 75. So it has a pore, when you pour inside, okay, barrel, from the barrel in the membrane, okay. because secondary structure of the protein, hydrophobic core of the protein, so it becomes goes into membrane and becomes barrel structure. So TOC75 can go in, so that transipeptide can go in through the TOC complex. Okay. Once it comes out TOC, then it has to enter the intermembrane space and then come TIC complex, TIC20. Okay. So think about it. Two membranes, two independent membranes, outer membrane and inner membrane. Look at me, okay? Not a slide. The outer membrane and inner membrane. TOC complex is here. No guarantee that TIC will be here. Same place, same point. Because they are all moving in the membrane plane, dynamic in nature. TOC is moving and TIC is also moving. Okay? Okay. So once it passes to TOC, TIC has to align with it. Then only can go through. Okay? So this alignment is done by TIC22 and a protein called POTRA. POTRA is an extension of that, but mostly TIC22. TIC22 binds to POTRA, who is the extension of TOC75. Okay? All right? All right? And TIC22 are present in the where? Intermembrane space. Do you see that? TIC22 present in intermembrane space. So this POTRA is hanging. Okay? Whenever sees TIC22, okay, bind to TIC22, okay. So transipeptide has come, moving in the along membrane plane, okay, like this. Then POTRA is just extending to the intermembrane space, and then to wherever you find TIC22, it binds. Once TIC22 binds, then it aligns with the TIC20 intermembrane space. Okay. So TIC22 is a very important component present in the intermembrane space, essential for protein transport. Okay. Now this TIC22, once these TOC75 and TIC20 align, okay, then you can see that it passes through the TIC20 complex. Okay. And then what happens? As soon as it comes out, it binds with CpHSP70. This is HSP70 here, CpHSP70. Chloroplastic, Cp stands for chloroplast. So chloroplast also, stroma, has also HSP70. We call it chloroplastic HSP70. Okay, all right. Binds to it is also molecular chaperone. Okay, binds to this. Then HSP70 binds to HSP90C. You have HSP90. There are HSP90 are many kinds of HSP90. So C, the 90A, 90B, 90C, and you bind to HSP90C. Okay, all right. Once you bind to HSP90C, then it binds to HSP93 and CLPC. What are these do? They are motors. One, their function here, as soon as it comes out of TIC20, okay, and there are these, you see these are anchored to the membrane, right? These guys, TIC110 and TIC40. So they are simply as anchoring it, anchors, staying there, okay. And these guys are attached to this anchors, okay. What are molecular motors? They start moving, okay. What do? They pull the transipeptide. Once it comes out, as soon as transipeptide docks, binds to CLP-HSP70, okay. As soon as it binds, these two guys, HSP90C and 93 CLPC, moves like a motor. Okay? When it moves like a motor, if you see, you know, it will pull it, right? You move it, you bind to it, and when it moves like this whole protein will be pulled back. Okay? So then this protein comes out. As soon as it comes out, the transipeptide is clipped by the stromal processing protein called SPP. Stromal processing protein. It recognizes where transit peptide ends and mature protein begins. Okay? All right? So transit peptide is clipped and then destroyed. Okay? You can say destroyed. Okay? And the protein comes out and folds into a mature protein. Okay? So what is it? Transit peptide. What is transit peptide? So a protein that must cross the envelope membrane, both the envelope membrane, reach the stroma, should have a transit peptide. And so transit peptide, for Rubisco, transit peptide is 20 kD. So total protein is 20 kD. 14 kD, as I told you, Rubisco small subunit is 14 kD. Okay? But synthesized at 20 kD precursor. Synthesized as a 20 kD precursor protein. Okay? So if you talk about precursor protein, so this is a pre-protein or PRSS, precursor of small subunit. PRSS. As an example I am telling. Okay? PRSS. So it has 6 kD transit peptide followed by 14 kD mature protein. Okay? You understand that? The 6 kD transit peptide is destroyed after it reaches the stroma, okay, as I showed you, and what remains? The 14 kD mature protein. So pre-protein and mature protein. So 20 kD is the pre-protein and 14 kD is the mature protein. Okay. After this reaches the stroma, okay, then LSU and SSU assemble. This is SSU, right? Small subunit. Okay. LSU is already present in the stroma, coded by the chloroplast genome. Okay. So on 7 days ribosomes. Okay. So therefore, then the protein assemble through molecular surveillance and all that. Okay. This is nutshell the overall process of protein input through the chloroplast envelope membrane. Okay. This is very oversimplification of the story. Okay. Okay, T236 super complex, we are not talking about it. T2 all together we call T236. Okay, but we are not talking about that. It's more confusing for you. Okay, therefore I am saying simplification of component. Will come. I'll show you later on. Okay, complexity of it. Okay, I'm just only showing the very simplified process. Okay. So when you talk about this, so number one, the protein, so transit one, the pre-protein or PRSS, precursor small subunit, must bind to the chloroplast membrane, right? Without binding, no import, right? The protein doesn't bind to the chloroplast membrane, there is no import. And where it must bind? And what is responsible for protein binding? How will you know that? Whether it bind to lipids or proteins. So how will you study this? What you have to do? First clone this gene of Rubisco small subunit along with the transit peptide, its own protein, all right? The whole genome, right? Rubisco gene, DNA, small subunit DNA. You have to clone it, right? Then next step is to do an expression vector. So you synthesize messenger RNA in vitro transcription, right? Then you take the messenger RNA, right? Centrifuge messenger RNA, then put into in vitro translation system. Okay. What do you do in vitro translation? You take this messenger RNA, put in a test tube. To this you add what? Rabid reticulocytes, or you can have wheat germ system. Okay. That will provide what? Ribosomes, all protein translation factors, and everything. Okay. All right. Ribosomes, right? Where the protein is synthesized, right? So you have to take, once the protein is synthesized in vitro translation, when you are synthesizing protein, you want to monitor the protein transport, right? Then you synthesize, let's say, with S35 methionine. S35 methionine. So you add, while doing translation, you add all amino acids cold except methionine, which is hot, which is S35 radioactive. Okay. So then your protein, wherever you have start codon, so it will go anyway. But wherever in between, where you have ATG, AUG, okay, it will be incorporated there. Where? Methionine. You got it? And let's say you have 6 methionines. Where? In Rubisco, small subunits. So wherever there is methionine, we have 6 radio-labeled methionine in the Rubisco small subunit now. LSU, including transipeptide if it is there. Okay. It will be there anyway, because ATG starts with start codon, ATG will be there, AUG will be there. Okay. All right. So you have now the radio-labeled protein. So now you have radio-labeled protein in your hand, right? With Rubisco small subunit along with transit peptide, which is radio-labeled now, right? Then you isolate intact chloroplast. You take isolate the chloroplast. You might have already done my practical. You can pass the Parker gradient, okay, to isolate intact chloroplast. Once you isolate intact chloroplast, add this radio-labeled what? Transit peptide to the chloroplast. Okay, take a test tube, take some chloroplast, add the radio-labeled transit, radio-labeled Rubisco along with transit peptide. Okay, all right. So now, priority for protein to import into chloroplast is that it must bind to the envelope membrane. You know it? So first actually binding. Okay, all right, understandable? So if it only binds to the chloroplast membrane, we just want to know how the chloroplast is simply binding to the envelope membrane or not. What do? You reduce the ATP content. You simply you need 1 millimolar ATP at least for protein transport. If you give 0.1 millimolar ATP, there will be only binding and no import. Okay. Why need ATP? This ATP hydrolysis, you see. To run this motor, you need ATP. Okay. If there is no ATP in the system, if you do not provide any ATP, this motor will not work. Okay. Therefore, only there is binding, but no transport into the stroma. Do you understand that? Understandable? Okay. So you give a 0.1 millimolar ATP, 0.1 millimolar ATP, very, very low ATP content you give. So that binding takes place, or you can add GTP also instead of ATP. Binding takes place, okay, but no import. Okay. And you then what you do? You did this assay, binding assay, this is called binding assay, right? Then what you do? You pellet your chloroplasts, okay, right? Again through a Parker gradient to the bottom, okay. Then wash it and take the chloroplasts bound to the what? Transit peptide and Rubisco small subunit, which is radio-labeled for you, okay. Then you run the gel electrophoresis of that, right? Add sample buffer, right? and then run the gel, okay? And then after you run the gel, you dry your gel, right? And then what do? Put Excel film over it, okay? Radio labeled. Then what do? After X-ray is labeled, X-ray. Wherever there is a band, radioactivity, you will see this, okay? Once you take it out, you will see that one lane you put will be your only translation product, right? The other is your what? Is your radio where you have done the experiment. So can you go switch to this blackboard? Can you switch blackboard? हो गया यही है ना while running your gel in vitro translation product. So this is your lane 1 is in vitro translation product. Lane 2 is your binding experiment where you did all those things, right? Right? And what you see? Both are in 20 kD. Okay. I told you your mature, the transit peptide is 6 kD. You know it. So what? So once 6 kD goes, if it goes imported into the stroma, 6 kD is gone. So how many remaining? 14 kD. So you will not see a 14 kD band here, right? Why? Because you do not do an import study. You did a binding study. Okay. A priori, that protein must be imported into the chloroplast. It must bind to the animal membrane. Okay? And that must happen. We must see that. Okay? Whether you get 20 kD band or not. Okay, that means your precursor protein binding to the chloroplast envelope membrane, to its receptors. Okay? Once you do that, then you do another experiment. Just increase the ATP concentration from 1 millimolar to 1 millimolar. Okay? Assay system. Okay? You take intact chloroplast. Okay? Add your PRSS. PRSS means precursor small subunit. You call PRSS. This is PRSS, precursor of small subunit, PRSS. Okay? All right? Understandable? So you take chloroplast, add your PRSS, okay? Add 1 millimolar ATP. Now there will be import. Okay? Then you do same thing. Okay? Then you do, after import, stop the import. Okay? All right? Simply put it in the ice bucket, it will stop. Okay? At cold 0 degree centigrade, import is going to stop. Okay? Then you stop the import, put the, whatever you want to do, gel electrophoresis. And if you do that, you will see, again this is PRSS, in vitro translation product. small band here. kD, this is 14 kD. You see that? You know it? Right? So, what is 20 kD band? This PRSS bound to the envelope membrane, but still not yet imported into the chloroplast. Still bound to the envelope membrane, are being transported to the what? Chloroplast. And this 14 kD band refers to those proteins which have already gone into the chloroplast, whose 6 kD band is chopped off, so you become mature protein, 14 kD band. Okay. This comes with autoradiogram. Okay, because measuring the tracing radioactivity only. So other proteins will not come here at all. It's a very clean system for you. Okay, to understand whether your translation product, or so-called precursor small subunit, is important in chloroplast or not. Okay. With this minimum assay, first you know that first you clone your gene, do a in vitro transcription to messenger RNA, do a in vitro translation to make a radio in presence of 30 methionine or tritiated leucine to get what? The protein, radio-labeled protein. Take this radio-labeled protein, then isolate it intact chloroplast, right? Then add it intact chloroplast to add this translation product to the intact chloroplast, right? Incubate it for 10 minutes, let's say, in presence of ATP, 1 millimolar ATP, then protein import, and you see the 6 kD band, 20 kD band is bound to the envelope membrane, and 14 kD band refers to those proteins who have gone into the chloroplast and whose transpeptide is already cleaved up by the stromal processing protein. Stromal processing protein recognizes where the transit peptide ends and mature protein begins. Okay, that function. Stromal processing protein. Okay, you see SPP is written there, right? SPP, right? Stromal processing protein. Okay, it is nothing but a protease, but simply clips at that point. Okay, that's it. Okay, it's called that we call a processing protein. Okay. So now let's say you want to know whether this protein, the receptor, is a protein or a lipid on the membrane or anything else, right? So what you do, you take the intact chloroplast, isolate intact chloroplast over a Perkel gradient. Okay, then treat with thermolysin, right? What thermolysin will do? Chew all the proteins present in the outer envelope membrane, but cannot enter the chloroplast. Thermolysin cannot enter the chloroplast. It is a protease which cannot enter the chloroplast, but destroy all the proteins, okay, which are present in the outer envelope membrane. Okay? So take the thermolysin treated chloroplast, okay? All right. Okay, take the thermolysin treated chloroplast, and then what you do? Isolate thermolysin treated chloroplast. Then you do import assay, binding assay or import assay. If your protein is if your chloroplast is treated with thermolysin, okay, then you do a import assay. Okay, then you will see a pores and the gel you are running. So this is your PRSS, which the band is like this. This is thermolysin treated TL, means thermolysin treated chloroplast, TLCHLP, thermolysin chloroplast, PRSS. But if you see here there is nothing, here there is nothing. No binding, no import. because all the receptors present in the envelope membrane are destroyed by thermolysis, right? So what tells you? Prior to this experiment tells you, that not so your receptor is not lipids. Membrane lipid is not working as receptor, number one. Okay, because you are being destroyed by protease, right? So they are not lipids, and the receptors are proteinaceous in nature. They are proteins, right? Isn't it? Okay. So from this experiment we knew that the proteins, the receptors are proteins. Okay. I'm not going details of this. Okay. But touching upon this. Okay. Okay. Receptors are proteins, not the lipids. Okay. Then next question is, there are many experiments went on and on and on. When coming out, I'm talking about the evolutionary discovery of protein import apparatus. Okay, went on and on and on. Then many experiments were done to see how the transit peptide, so we do not know which protein is binding, right? So what they did, they made some anchors, or flagged. The rubisco transit peptide was flagged by, and there are some added some binding motifs, extra binding motifs. Okay, and then this radiolabeled protein, and this will bind to the protein wherever it goes, right? I will not go details of it. We will bound to it. Okay, and then they found out to which is cross-linking. So they are simply cross-linking the transit peptide to the protein present in the membrane to find out which protein is going to bind. Okay, let's say it is binding to TOC 159. Okay, covalently bound now. Okay, we made a covalent bridge between the how we made that, all that I'm not going to speak about right now. We don't have time for it. Okay, 159 and what, 20 kD protein, right? Now the protein that comes out, radiolabeled protein, because rubisco is radiolabeled. Okay, radiolabeled protein that comes out is not 20 kD anymore, but 159 plus 20, 179 kD. Okay, they found out that the protein is now 179 kD. Okay, that means 179 minus 20 is 159 kD protein it is binding to, right? So they thought that probably this is the receptor to which probably is not completely confirmed that. Then what they did, okay, they cloned 159 gene and silenced 159, TOC 159 by mutation. Okay, so TOC 159 got silenced. So this plant does not have TOC 159, right? By mutations. Okay, they got a mutant which doesn't have TOC 159, and they found that this is very yellow, doesn't green, mutant doesn't green, have very little chloroplast development. Okay, and the plastids that isolate from it could not import the chloroplast. They end up bind to the PRSs, precursor small subunit. Okay, many kinds of experiments confirmed. Okay, that TOC 159 is the receptor of the transit peptide. Okay, there are many ways, many things that have, whether the protein is imported like this, or previously we are thinking the protein is going in 3-dimensional form. 14 kD protein, 20 kD protein is entering both the environment in 3-dimensional form and reaching the stroma. We are not thinking before that is being threaded in. This is threaded in, right? We are thinking the protein is being imported in 3-dimensional form to the stroma. Okay, then our knowledge, etc., changed. We did many, many experiments, prove or disprove our concepts. And later on we found that it is going threaded in rather than going threaded in. I cannot give all the examples here, all the experimental verification of it right now. I have only 1 hour lecture, okay? So, similar for all the components we talk about here. It's not that easy to say, okay? Many, many experiments went, many people came out, many proteins, different proteins. Finally, certain proteins have stood the experimental test. But I'm telling you, don't be biased by it, okay? We'll tell you in a few moments, there are many issues on this concept. But I give you a consensus concept, okay? Or the proteins associated with the membranes, okay? But tomorrow in a textbook or after a few years, you will see this concept may not be there. Some other membrane proteins will substitute it. Look, can you go back to the blackboard? The boy is gone. To the yeah, that's the one. Oh, good. Top. Yeah. So all these proteins you see, so therefore when somebody take 236, okay, I keep it to be 236 as a complex structure, okay. We'll talk about it if time permits, but I don't think there's time for it. Okay. Now you see. What is this? Transit peptide. I gave simplistic picture. Once a transit peptide, they want the protein synthesized, right? With this transit peptide. Then it binds to many components. One component is 14-3-3 complex. 14-3-3 protein. It is a class of proteins, okay? Also is a molecular chaperone. Functions as a molecular chaperone. It keeps the protein in transport competent for. As I told you, if this transit peptide, this protein, does not bind to proper what? Molecular chaperones, then it becomes transport incompetent. Means it cannot cross the chloroplast membrane. Okay? So we call transport incompetent form. Okay? You see, I have given a topic unsolved questions. You see my title they have given to me. Unsolved questions of Elmer transport membrane. Before I talk unsolved question, I have to tell you what is solved question first. What do we know? Okay. Now this is RBCS, rubisco small subunit, right? This band HSP70 and 14-3-3 proteins. And this is going to the import apparatus, TOC and TIC. Okay? We put TOC 75, the input barrel, TIC 20 also another input barrel. Okay? Crossing the inner membrane, barrel protein. Okay? This is also a concept. This is, we are putting TOC 33, TOC 159, right, as receptors. Here is TOC 132. Do you see that? 159 and 132. Okay. They also, the TOC 132 also work as a receptor. We say, we'll see later on, that TOC, the two sets of protein, one is photosynthetic proteins and the non-photosynthetic proteins. That enters the chloroplast, right? The rubisco small subunit photosynthetic protein. Okay, all right. But there are many proteins that goes in, which is a non-photosynthetic function. We have some other functions, but not in doesn't work in photosynthesis. Okay, so photosynthetic proteins goes through input apparatus like this. Non-photosynthetic protein goes in input apparatus like this. So depending upon which, if you only bind to HSP90, okay, then also there is protein import. Here there is only no protein import, but there are many other stories come about it. But we will not go, this is another component could be E1A and Y and all that, don't bother about details, okay. So it's a various cytosine binding proteins and import competence and import apparatus. Okay. First is the various cytosolic binding proteins, whether 1433 protein, HSP70 or HSP90. Okay. On another protein called Y, we don't know its chemical nature very well yet. Okay. And they all bind to it. So the complexity starts here. Okay. So it is saying import competent. What is import competent? So it binds to the protein and goes to chloroplast. It does not bind to the proper import proteins, proper chaperones in the cytoplasm. Then, though all these transit peptides are ubiquitinated, okay, and go to the proteasome complex and getting destroyed. You see that? Okay. So transit peptide alone, okay, does not guarantee protein import. Okay. All right. Many proteins who do not bind to the proper molecular chaperones present in the cytoplasm, they get destroyed. Okay. I will keep these slides for you. Yeah. Now you see. We are saying, what is this? So these are resting, this is import apparatus, resting stage. Right? Okay. Now transit peptide comes along protein, is not showing you the molecular chaperones binding to it. Transit peptide is there. Now once it binds, what happens? They all are bind with GTP. Top 115 bind with GTP, and top 34 is bound to GDP. Do you see that? For resting phase, top 115 is bound to GTP and top 34. Okay. Both of them, as I told you, work as the receptors. Okay, 115 and top 34, they bind to GDP. Okay. Now, once it binds, transit peptide binds, you can see it is bound to it. And next step, the GTP hydrolysis, right? GTP is GDP, right? Then one GTP is last. Then you have to add GTP. So, your replenish GTP. Okay? GTP hydrolysis took place. So again, you have to put back GTP, give GTP. So, okay. So now see, this is bound to GTP, and all of them bound to GTP now. Once you get GTP becomes binds. What you, in essence, is the GTP hydrolysis. Okay? That determines the binding of the protein. GTP must bind, that's with GTP. GTP should be hydrolyzed, okay, to do the initial protein binding and import through the intermembrane space. Okay. Ready? Okay. So now it's entering the where? In the tick 20. Okay. If you give GTP gamma S, what is GTP gamma S? So guanosine, guanosine, guanine, base, base, sugar, guanine, then sugar. Then you have three phosphorus. Last phosphorus is the O bond, you have S bond, sulfur bond. So it is non-hydrogeable. So it cannot hydrolyze to GDP. But GTP gamma S is the compound of GTP which is not hydrolyzed to GDP. Okay? If you get GTP gamma S, there will be no GTP hydrolysis and protein binding import will stop. Okay, I have just finished this. So as I told you, there are complexity photosynthetic proteins, non-photosynthetic proteins. Okay, so different complexes. Non-photosynthetic proteins are TOC34, TOC132. For photosynthetic proteins, TOC159 and TOC33 and TOC75. So TOC complex TOC complex remains same. TOC complex discriminates between photosynthetic proteins and non-photosynthetic proteins. Rubisco will pass through this process with a small subunit, or plastocyanin, or any other protein, okay, that goes through this pathway. Okay. Whereas non-photosynthetic proteins will pass through TIC32, TOC34, TOC75 complex, and they both go, and TIC complex remains same for both of them. Okay, they go into the chloroplast. Okay, since your next speaker is ready, the relative abundance of translocation isoforms appears to critical and balancing port distinct classes of pre-proteins required for protein remodeling during a plastic type transitions or response to physiological changes. Yeah. Do it, this is pre-germination, the post-germination, you can see protein transport in etioplasts, protein transport into chloroplast. How it is changing. Okay. All right, we stop here, right? Any questions you have? Any questions?
What we'll do, maybe during lunchtime you can talk to him because the other speaker is here. Now he'll be here during lunchtime. You can talk to him, discuss with him.
Yeah. Okay.
Is it okay, sir?
Yeah, sure.
And I would like to thank again for coming. And I think he's leaving today evening. Okay. So big thanks to you, sir, for always helping.
Thank you.
And we have long association with him, almost 40 years.
Right, several years.
Several years. And where we both did PhD with we have commonality.
Yes.
The first student of Prasanna Mohanty and the last but one student.
Yes.
So thank you, sir. Thank you for your excellent talk.
Yes.
And they will interact with you during lunchtime.
Yeah, okay. All right. Thank you.
was in Kerala state last few days and also met other Indian students, and I enjoyed a lot by communicating with them. So today, once again, I'm happy to be here back, and if I have a more interaction between us. Okay. So this is I'm going to talk about the chloroplast genome, but more about how we can manipulate chloroplast DNA. So, sorry. So as already I introduced this, I'm mainly focused on the chloroplast of green alga, Chlamydomonas reinhardtii, because this organism is amenable to a chloroplast transformation. So this organism is not only chloroplast but also nuclear, and as well as mitochondrial genome can be transformed. So I just remind of you the structure of this cell. So single cell, nucleus, and the chloroplast. And the volume of this chloroplast is about 60% of cell volume. And something like this. So you can see this is on microscopy. Looks like the flagella is moving around, and then chloroplast is and cytokine membranes present, and this is pyrenoid. So it contains a lot of rubisco protein here, and also starch. So the chloroplast DNA is present in the stroma of the chloroplast, forming a nucleoid complex with DNA and the proteins, and some are loosely or probably loosely associated with cytokine membranes. So this is a circular DNA of the chloroplast genome, about 5,535 base pair, and here and here, bold line, it's inverted repeats. The same DNA sequence repeated, duplicated, but in the opposite direction. Okay, and this is a small single copy region and large single copy region, and some genes like PSBA here, its green part is exon, and the gray part is introns. Some chloroplast gene has introns, and also you may not see some genes. This light green is photosystem 1 encoding the genes encoding photosystem 1 subunits, and dark green gene is encode a photosystem 2 subunits. And also blue is encode a subunit of cytochrome B6F subunits. And this yellow is genes encoding subunit of ATP synthase. So 4 major complexes are present in the cytokine membrane. Some subunits are encoded by chloroplast. And the subunits encoded by chloroplast is very often here the very essential subunits containing catalytic sites. And also, this orange is rubisco ribosamides. And others, gene expression involved in gene expression, RNA polymerase, the ribosome proteins, and other genes are involved in some PS1 assembly or chlorophyll synthesis protocol fluoride, oxidoreductase, something like that, tRNA and ribosome RNA. So these genes present in the chloroplast, and maybe its total number is usually less than 100. And the chloroplast usually has, it is estimated that about 3,000 or 2,000, maybe 3,000 proteins are present in the chloroplast. So only a small number of proteins are encoded by chloroplast gene. But for the photosynthesis reaction, the chloroplast encoded proteins, there are many very important proteins as a function. So I just today introduce chloroplast transformation. So as you can see here, the chloroplast is inside the cell, surrounded by two layers, cell wall and cell membranes, cytoplasmic membranes, so very hard. And the chloroplast is surrounded by two membranes, outer envelope membrane and inner envelope membrane. So DNA is here. So if you want to transform the chloroplast DNA, you have to deliver the DNA into the chloroplast. But there are at least four layers that prevent the introduction of DNA from the outside. So what we use is a particle gun to deliver DNA into chloroplast. So the chlamydomonas has the diameter is about 10 micrometers, and we use a tungsten particle with a diameter of about 1 micrometer, much smaller. And then we coat this particle with transforming vector DNA. Then shoot the chlamydomonas cell with particle gun at the speed of like a sound, 300 to 400 meters per second, very fast. Then this particle may block the cells like this, make a big hole, and then the cell will die. And maybe some cells may not be hit by particle. And fortunately, or I don't know, unfortunately for chlamydia, tungsten particle it transferred into the chloroplast, and the tungsten particle remain inside. Then the DNA coated on the surface of the particle will dissolve into the chloroplast, and then you can introduce the transforming vector into the chloroplast. So this is the particle gun we are using, and this is a controller, and this is a chamber. We make this inside the chamber, the vacuum. Okay, and then particles are shoot using the power of helium gas. Okay, very short pulse, helium gas pulse, shoot. And then inside on the agar plate, chlamydomonas cells spread on surface or agar plate with a monolayer. And then if the inside of chamber is vacuum, the small particle will not be accelerated so much, so very at high speed. You can shoot the cells with this particles. Okay, so this process is not difficult; it's easy. So this is how it works. So we on the agar plate, we spread cells like this. Basically, if you use a fixed concentration of cell and also you add optimize the volume, it's not difficult to spread the cells as a monolayer on the agar plate. And then you shoot the particle. Then we have a selection system. Now, just after shooting, and then 1 week or 2 weeks later, you will see colonies like this. These are transformed. In the middle of the particle, the strong gas spread, blow away the cells and kill. So there was no, there's no transformed colony. So in the surrounding region, the optimized condition, you can see many colonies. The question is how you select these transformed cells. Is that we use a selector marker cassette that confer the cells antibiotic resistance. So we use the bacteria AADA gene that encode the enzyme that inactivate the spectromycin. That is, spectromycin is specifically block the protein translation in the ribosome of the bacteria type ribosome, not nuclear eukaryotic type ribosome. That is ATS. But this blocks the bacteria type 70S ribosome activity. So Chloroplast has a ribosome of bacteria type ribosome of 70S. So this selector marker blocks the translation in the chloroplast, but with this selector marker, this inhibition, the block is removed. Okay, so with this,
we will add that antibiotic after the gene content.
Yes. Okay, good question. We already add the spexomycin on this plate, or after transformation, after shooting, we add the spexomycin. So answer is we use the agar plate with spexomycin. But we plate this, and within 1 hour, we shoot. Okay? So this short period, because we choose the concentration of spexomycin properly, so it does not immediately kill cells. Okay? So it's easy. Now you just shoot and put it on the incubator, in the incubator, and then you don't need to care. Just 1 week or 2 weeks later, you come back and then see, oh, there are many colonies. So then you will be happy. So AD cassette, the bacteria type, is also a codon usage optimized for the bacteria. isolated this gene. But in this case, we can use AD cassette, of which codon usage is optimized to chloroplast of chlamydomonas chloroplast. And then only coding region will not be expressed in the chloroplast. This AAD openly different coding sequence is fused with 5' ATPA gene. It's a regular 5' region of ATPA, chloroplast ATPA gene. And also, this is fused 3' RBCL gene. Okay, so this region is very essential to regulate transcription and translation in the chloroplast. And this region is also very important for stability of messenger RNA in the chloroplast. But this region can be deleted. We know that it's possible. But because we don't know, the efficiency may decrease or not, increase, probably decrease. So usually we use this. But in a specific reason, we don't want to use this 3' RBCL. So then we can remove this part. Okay, this part is not essential. So this is the first example of the heterogeneous expression, foreign gene expression in the chloroplast, and it worked. Okay. And then what happened if you introduce a transforming vector into the chloroplast? This is an example of targeted knockout of the chloroplast gene. So this is a host chloroplast DNA, okay? And this is a transforming vector, clone in the plasmid. And we clone first this target DNA. This is a target DNA we want to of interest. The upstream and downstream region, some region about 0.5 kb or 1 kb, certain size. We subclone this DNA into the plasmid. And then we insert in the middle of the gene AD cassette, spectromycin AD cassette. So that gives us a spectromycin resistance, okay, the white part. And then we introduce this transforming vector. So this homologous region, the red region, and blue region, the crossing occurs. So then this was replaced by this region in the transforming vector. So after transformation, AD cassette was introduced into this gene in the transformant chloroplast DNA. This reaction is enhanced in the presence of spectromycin. In the absence of spectromycin, this homologous recombination does not need to occur in the chloroplast. But in the selection condition in the presence of spectromycin, this recombination is enhanced. Otherwise, cell dies. Okay. So then this is insertional inactivation of the gene. You can knock out a gene by insertional inactivation. And or you can, if you knock out the gene of your interest, or you subclone the gene, and then you delete, or almost all the part or part of the gene coding region or all region. Okay, and then after deletion, you insert the AD cassette. Okay, like this. But between this homologous recombination, this happens. It's deletion mutant. This is insertional. This is deletion. But it contains AD cassette. Otherwise, this does not occur completely. Okay. This is mainly, you can knock out the chloroplast gene. But there's some gene that is essential for the survival of the chlamydomonas. So you cannot knock out. For example, the gene involved in the gene expression of chloroplast gene expression, you cannot knock out. There's some limitation. But photosynthesis gene can be easily knocked out because chlamydomonas can grow heterotrophically in the presence of acetate. Okay.
So CRISPR we cannot use in CRISPR-Cas we cannot use for chloroplast genome.
Sorry?
CRISPR-Cas
no, you don't need. This is easy. Nuclear transformation you can use, but this is you don't need at all. Very specific because homologous recombination occur like a bacteria in the chloroplast.
Sir, which vector is specifically using for this? Which vector is using? Bac vector or yak? Bacterial vector or?
Bacteria?
Which vector is using specifically any?
You say what the vector used?
Vector, but not bacteria. No, anything.
Anything you can.
Yeah. Blue script or no, pack or SPAC is not very important. But for cloning, the high copy number plasmid is very easy. So usually we use the blue script for this. So then another the method is to site-directed methylation. You want to introduce a point mutation to change the codon or add the amino acid sequence into this. Like if you fuse the tag to the protein of your interest. Or you can delete some specific residues, something like that you can do, or insert in the middle of protein. So in that case, the transforming vector you subclone the same way, and this is a gene of your interest, and then you insert AD cassette downstream or upstream of the gene of your interest. And the position of the insertion does not affect the expression of this gene. No. Okay. Otherwise, the insertion of the AD cassette decrease the expression of this gene, so that affect accumulation of protein. So you need to find this position in the chloroplast. And sometimes there are many genes clustered. So you don't find the site to insert the selectable marker very close to the gene. So sometimes you have to insert it a little bit distant. So that happens. And also, if you don't have any region, so then sometimes we can make two transforming vector. One is this selectable marker at the site, as already you know, that does not affect any gene expression, and also vector that has a point mutation. And these two transforming vector mixed and then transform together. So co-transformation we call it. Then you select first the resistance against the spectromycin. Among the transformants growing in the presence of spectromycin, you screen the clone that has point mutation. So this is you need a little bit more work. And it depends on the experiment, but sometimes you have a positive clone that has a point mutation in, I don't know, 50% or sometimes 10%. But you can screen the tens of colonies, right? Tens of, 10, 20, 30, 40, 50, still possible. Then this, you need a longer homologous sequence here to insert. So the homologous recombination occurs, then both genes, mutated gene and the cassette are introduced into the transformer chloroplast DNA. So this is a way. Because then before I talked about the insertion of tag to the chloroplast encoded protein. So this insertion was used this method. So I'm going to show our first knockout of gene, chloroplast gene, PSHC gene that is encoding iron sulfur protein of photosystem 1. This is one of the first chloroplast gene knockout. So PSHC gene is located here, single copy region. Okay. Yeah, this is the same thing. Okay. So we cloned this subclone, the DNA fragment containing a PSHC. So at that time, for example, it's a little bit technical. This is large EcoR1 fragment, and then subcloned PST1 and SAR1. Okay. This is an old experiment. It's published in 1991. So at that time, not so many technique. PCR was just commonly used. So to do this, by chance, this PSHC gene is small gene, but we found that NCO1 site is there in the middle of PSHC gene. And NCO1 site is here also. So we clone the PST1 and SAR1 site, or XBA1 and SAR1 site. Then there is no, only NCO1 in the middle of the PSHC gene is a unique site. Okay, so we clone it into plasmid, PST1, SAR1, or XBA1 and SAR1, and then digest this plasmid with NCO1 restriction enzyme, and then insert it AD cassette here, ligate it. So the making plasmid was relatively easy. Okay. At that time, I was first engaged in molecular biology. So for me, it was a little bit difficult, but relatively easy now. It's very easy now. At that time, restriction enzyme was not so good, and then some enzyme didn't digest the DNA so well. That kind of things happened. Okay, then we transformed this plasmid into chloroplast. And then we checked the transformant. Selected the cassette spectromycin in the presence of spectromycin, and then putative transformant. Until we confirm the DNA, we can't say it's a transformant, because sometimes a spontaneous mutation becomes spectromycin resistance. Okay. Then at that time, now we use a PCR But at that time, we used the Southern blotting experiment. Do you know the Southern blotting? Yes? Recently, only few people use Southern blotting. But for now to analyze this, the transformer, just we need to amplify near PSAC, and then if there is an insertion, PCR product becomes larger. Without the insertion, PCR product is like Y type. It's easy. But at that time, we have to analyze the Southern blot analysis only. PCR reaction was sometimes very tricky at that time, so, and also expensive. So the oligonucleotide synthesis was also not cheap. So we used Southern blotting. So we digested total DNA with what is EcoR1 and SBA1, so EcoR1, XBA1 here and here, from here to here, okay? Here to here. And then separated a fragment of DNA by agarose gel electrophoresis, and then blot it onto the membrane, and hybridized with a probe of this region, okay, with labeled with phosphate. And then we detected by X-ray film, and then developed, and then the fragment hybridized with radioactive probe was detected. Okay, this is Southern blotting. And in the wild type, here, its size is 3.0 kilobase pair, KBP. But if PSAC has an insertion of 1.9 kilobase pair cassette, so the size increases to 4.99 kilobase pair. Okay, don't worry about this additional one. So at that time, somehow something happened. So as you expected, this is a probe with PSAC gene. So 3 kb signal are upshifted for 4.9. So it shows all DNA are transformed. Chloroplast DNA are transformed. Okay. And we also hybridized with this AD cassette, the probe hybridized with AD cassette. Then there's no signal in wild type here, but signal at 4.9 kilobase pair appeared because this contains PSAC and ADA gene. So we confirmed that, oh, we can make. We made it. Okay. Then what we will do is analyze the phenotype transformers. We did another analysis, the messenger RNA level. So in the wild type, the messenger RNA, the probed with PSAC gene, give us a 0.45 kilobase messenger RNA and 1.1 kilobase messenger RNA, 2 messenger RNA. But in the transformant, we couldn't detect any transformant, so transcription didn't work or very unstable. The messenger RNA becomes very unstable. So the AD cassette messenger RNA, there is no in the wild type, but in the mutant, there's signal. So AD cassette is transcribed and probably translated because the cells are resistant against spectromycin. Okay, so today I'm not going to talk about these two, but just I tell you that we did mapping. So point 0.5 is starting here and then end in the downstream of PSAC. So this transcribe on the PSAC gene. And larger one is the starting is the same, but it goes through a little bit downstream. And then we found the open reading frame downstream of PSAC at that time. Then that was encoding photosynthesis protein we found at that time. The whole genome of chloroplast was not have not been determined. So maybe if I have time later, sometimes I talk about this. So the phenotype, another phenotype, the activity of the transformant. We measured the fluorescence induction kinetics. I think you did some laboratory course this time, PAM measurement, okay? So this is a chlamydomonas fluorescence induction kinetics. It's time about 2 seconds. Then you illuminate the cells with actinic light, fluorescence go up a little bit, how do you say? Strange kinetics and the peak, and then go down. Okay, this is Y type. Without killing the cell, breaking the cell, you can measure this kinetics. And 4B2 is one of the PSAC knockout mute transformant, one of the clone. Okay, so kinetics like this, it's exponentially go up and then stay. It's completely different. So you know from your laboratory course that what this phenotype means. Did you try, experience something like this? Tell me.
Speak loudly.
You want to speak microphone?
What did you ask? We are asking what did you ask?
What did you ask?
Can you repeat the question?
This is Y type kinetics, and this is transformant. So but this difference in the kinetics of fluorescence induction kinetics, what this means? Absence?
Fluorescence in mutant is more than wild type.
Sorry, I don't hear well.
Fluorescence in mutant is more than wild type.
Higher. Yeah, obvious. But what this suggests? You can explain why it's higher. Did you do this kind of experiment? Yes? Okay, why is higher? So, okay. During illumination, the fluorescence yield increases like exponentially. This means photosystem 2 activity is there. Okay? So it goes higher and it didn't go down. Okay? So this means that the fluorescence is basically emitted from photosystem 2. The photosystem 2 acceptor QA or praskinon pool is reduced, fluorescence yield increases. And this QA or praskinon pool is oxidized, fluorescence yield decreases. Okay, so this means the wild type, it's very complicated kinetics. PS2 is reduced, but certain point it starts to oxidize. because photosystem 1 extract electron from PS2. So oxidize plastiquinone pool and oxidize acceptor of PS2. So this means PS2 activities here in the Y type and also PS1 activities here. But in the mutant, it's go up. So there's no activity to extract the electron from PS2 to from PS2 because on the PS1 activity is absent, so electrons accumulate. So PS2 is highly reduced. So the fluorescence yield becomes highest. So it happens higher, and they didn't decline. So this indicates that PS1 activity in this mutant is absent. This is as expected because PSSC encodes iron sulfur protein of photosystem 2. This is essential for PS1. So this is as expected. But we can confirm easily by measuring fluorescence induction, the phenotype. Then we expect the PS1 protein is absent or present, but not functional or functional or non-functional. So we just simply analyze the protein polypeptide of thylakoid membrane. We isolate thylakoid membrane and analyze the polypeptide pattern. This is Y type. PS1 reaction center is around here, and some proteins are PS1. Okay, there are many. And number two is negative control of the mutant. Already we know the PS1 is absent.
The acceptor side is there, so the oxidation is affected.
Yeah.
So, but why reduction is also maximal in mutant?
This is the balance between the reduction and oxidation. Okay, and also it's always this is also balance of redox between the PS2 and the PS1. So
In the mutant
there's some explanation, but I'm not quite sure if they are correct or not. But this is also reflect because PS1 and PS2 activities start at the same time if you put on the light. So always, the equilibrium occurs. So in the absence of PS1, this kinetics is totally absent. But in the presence of PS1, there is some
okay.
the balance is
The energy distribution will be
energy
balanced.
distribution or the balance of the distribution of electron among between the PS2 and the PS1. But I'm not quite sure. There's some explanations, but it's correct or not. Sometimes I doubt some explanation. But this kinetics is always observed in the Y type, but even the PS1 and the B6F deficient mutant is same like this. This disappears. So this is always appears in the presence of PS1 activity. So PS1 in transformers almost absent here. Okay. So although we knocked out the PSAC genes that encoding a small subunit of PS1, but other PS1 protein were also seem to be absent or decreased. Then we tested reactions, the immunoblotting analysis. At that time, the sensitivity of immunoblotting was not so high because we didn't have the technique of enhanced chemiluminescence ECL method. At that time, ECL was not, we didn't know. We still had, how to say, this was used isotope to detect high sensitivity. It was a little bit scary because iodine 125 is very strong gamma ray, so I have to work in lead metal, so heavy. So if you use the radioisotope emitting beta ray, it's okay, but the gamma ray is very powerful, so it penetrates. So if you want to shield yourself, you need a heavy metal block. So it's not easy to work. And PS1 deficient mutant completely lacks PS1 reaction center protein, but it a little bit accumulate in the PSAC mutant. It's very much reduced. So immunoblotting is more sensitive. So other small subunit is totally absent. So the question arises why PS1 is other PS1 subunits are reduced in the absence of PSAC. This means that the knockout in PSAC gene or translation affects the translation of other PS1 subunits, block or suppress the translation, or synthesized PS1 proteins unstable and degraded. So to analyze this, we did a pulse chase labeling experiment as I talked before. Okay, we used this S35, I think, at the time. So we labeled, and 2, 5, 8 is the labeling, pulse labeling, and 3 and 4 is chase experiment. So this was reaction center protein, and the wild type pulse labeled protein was stable during chase experiment. Okay, and this PS1, no, not really. This is only chloroplast encoded protein. And PS1 minus mutant is also pulse labeled. It's a little bit strange, but these are not really PS1 because it's upshifted. And PSAC mutant is pulse labeled like this, and during a chase it disappeared. So this means if you knock out PSAC, reaction center protein, other PS1 subunit, the main reaction center protein, synthesized but degraded rapidly. So if the synthesis rate is the same but degradation rate is faster, so steady state level of accumulation of PS1 is reduced like this. Maybe 20% or 10% of wild type level. So this means that PS1 complex is unstable in the absence of PSAC. Okay, this is a example of knockout of chloroplast gene. So this is first experiment for. So we just knock out already function the gene of which product function is known. Okay, because we needed to test the phenotype. So without unknown, if you work with the gene that is of which function is unknown, in the beginning it was not easy to analyze the phenotype, so we choose this gene as a target. Okay. So I think next is the point mutation, but its time is running out, so I think tomorrow I will talk about introducing a point mutation. And so I will stop this talk. So I welcome your question and comment or anything.
Yeah, you can discuss with him during lunchtime. Maybe they're all hungry now.
Sure.
So
Okay.
today is special food, biryani.
Biryani, okay. So you need you need more energy to discuss after eating the biryani. You're welcome to ask any question there. Okay, thank you. For your
Thank you. What? Hello. Hello.
She's starting.
Let's start. All are tired with biryani? Okay, we will keep it simple. So from like many days we were talking about Blue Native page already. So now you people know what is Blue Native page. Even that day thylakoid isolation, I explained you why we do Blue Native page and all. And sir also explained you what is Blue Native page. So can someone tell for what we are doing Blue Native page, why we are not doing SDS page? You need intact protein complex, which is photosystem 1 complex and photosystem 2 complex. We need the complexes for studies, so we do Blue Native page. And we want to see in super complex level there is any dissociation, so that the bands will be lesser in intensity or more, whatever the band intensity, at least you can tell PS1 is more, PS2 is more, what is happening with the photosystems. So this is the basic thing why we are doing Blue Native page. And here we won't use any denaturing agent, no SDS, no beta mercaptoethanol, no heating, nothing. Everything will be in ice cold temperature, and also you are going to work with complex, so it should be in dark. Whatever I told thylakoid membrane isolation, Blue Native page also it will be very good if you do in a cold room in a dark condition. Then okay, in SDS page what is the buffer we use to keep the pH? TrisHCl. Here we will use little bit more more refined buffer system, which is Bistris and Trisin, and pH 7 should be maintained in both. 7, right? Not 7. pH 7 should be maintained in both. In SDS page you will have different pH, right? But here you will have same pH, and pH is very important because to maintain the voltage, to run the complexes, this pH will play a key role. So pH of whatever the two buffers, you start BN page first by preparing these buffers. So when you are planning to do a BN page, first you are going to prepare two buffers, which is like Bistris and Trisin. Okay, and the Bistris will be part of all other ingredients or the composition of the gel preparation. Okay, and here you are going to have a gradient of gel, not like in SDS page you will have a single percentage, right? 13% gel, 15% gel, whatever. I think here if you see in SDS page there will be different percentage of page, and it will be fixed in whole system. But in case of Blue Native page you will have a gradient of gel, which is like according to your complex size you can change. Sometime it will be like 4 to 12%, sometime you will vary a bit 3.5 to 12%. These all the percentage will be depend on which complex you want to separate, and depending on the organisms also this will change. Which one? The gradient preparation I will show you. We have a gradient mixer here, and you can't prepare it like in SDS page. You need to make a gradient. So if you just add something with a pipette, you are going to you are not getting a gradient there. Everything will be mixed. So here what we will do, we will have a higher percentage, which if in our case we are going to make a 3.5 to 12% gel. So I'll have the minimum one 3.5%, and we'll have the heavy one as 12%, which is called light and heavy. Okay, this what are the extreme you are going to do. So this percentage, and what makes the percentage of a gel? What you are telling percentage? Like if it is a 15% SDS gel, what is that?
Is in percentage, what the percentage of is dissolved in
No.
smallest pore.
No. Okay, pore, which is related to what is making the pore? Acrylamide. So acrylamide is making this pore size, right? So this percentage basically making the pore size. So which is related to acrylamide. So you change the acrylamide concentration accordingly to make this 13% or whatever I am going to make, you are going to change this only. Your heavy and light you can vary. So first you will try a normal one. So you understand, okay, in this percentage mine is not coming, I need bit more pore size. You decrease the or increase the acrylamide percentage. This is how you make. Okay. And for next what? Okay, we prepared the buffer. Now what all we need in a BN page system? Can you get my copy? Here or here? Here I think, right? So we talk about the pH now. We talk about the gradient. And if you want to read from the basics to more, refer to Eva Marie Arrow paper. Arrow. EVAMARIARO. And they will have a video tutorial in that paper. If you go to their website, they will have a tutorial how they make the gradient and everything. Because if I start BN preparation today, I'll make buffers. Tomorrow I will make the gradients, and next morning only I will do. So we can't do this possible like a 3-day experiment. So I'll try to give a maximum idea about what we are doing, everything. Now you can go and see this video and read the article. You will get more idea about how to separate and all. Let's go to that mixer and I will just try to explain how we are doing that. Take that also. That. Again we can't. We'll just go and come. It's fine. Maybe 5 people come with me. I'll show what is. Nothing is there. I will just show you this. Maybe first 5 people come with me, or half of you people can come. Yeah. So, see, I wrote here heavy and light. So, what you're going to do, you will connect this pump. This is basically a pump system, so that it will go, you can control the flow here. So, you don't want to just mix everything. You want to go it very slowly so that it will form a slow gradient. I already made this from morning. Otherwise, this gradient making will take again 3-4 hours. So, I came in the morning and I put this because it's not feasible to do this. So, I just connect this to this using a small tip or something. I'll keep it here and I put the different percentage of solution, light and heavy, separately. You should not mix it. It should mix slowly from here. From here to here there is a connection. So, according to that, here you can maintain the flow, speed, everything. Maximum slowly you can do, like here I keep almost 2.3 ml per minute. 1 minute only 2.3 ml will go inside. So, this is the flow rate. If you decrease it to 1.5, 2, it will be more good gradient. Slowly something is going, then it will form a very nice gradient, right? So, that is what why we are using a mixer. You are not going to put using a pipette. So, that work will be done by this mixer machine. So, it goes like this and slowly this will control the flow rate. Sucking and flowing rate will be controlled by this mixer machine. That's it. And once you fill up to here, you are going to add normal like isobutanol or whatever, and then after 3 or 4 hours you can see it will be solidified and a nice gradient will be formed. And here APS and TEMET percentage will be very less, not like SDS page in half an hour it will solidify. No, the solidification or the polymerization should be very slow so that the gradient will be formed very nicely. So this is the concept behind this. So we'll keep it, sometime we just keep overnight like that so that it will form slowly, and next day only we use this. So once it is done, like a normal SDS page, you can prepare the stacking and load. There APS and TIMID we will be adding more so that in half an hour stacking will be done. So now I just poured the stacking. It will be ready by the time we prepare our sample. Any doubt related to this?
Stacking and dissolving should be separated.
Yes, because you need the comb, right?
Yes.
So pH and all will be same. Every composition will be same, no difference.
Machine only the resolving is
Only resolving, because resolving only you will have a gradient. Yes, you can do. It's clear about why we do this mixer and what we are clear?
Below the
Yes, yes. Maybe you will keep always a like space between the resolving and this, right? So at least a 0.5 centimeter here gap will be there. Okay. So maybe next people can come.
yep. The mixing, you mix both the heavy and light, and then it is
No. I will open this pump. Then it goes like this. Then I will open the light, because you need down the heavy 12%. So you open it, 12% goes slowly. Then you slowly open this. Once it is reaching here, so here they will start mixing. So now whatever it goes will be slowly mixed thing. But once it is going by drop, it will go.
first
How it
heavy
will be? Something heavy and light. It will go drop by drop, and you are keeping a 4 hours. Obviously, the heavy things will go down and the will come up, right? So that's what here happened. And also to make this heavy, we will add glycerol in heavy. It will be viscous. So with that glycerol heavy, it will automatically go down. And here we won't add glycerol in light. So that's how this system works.
reference what I've given, they have the protocol also.
Yes, they have the protocol and they will show how they are doing. There is a video. If you click, you can see that as well. Okay, can you ask the other people to come? Remaining people can come now. So as I told, we are not going to do something with a pipette like SDS page. We need a very nice gradient from 12 to 3%. So I told you a higher percentage and a lower percentage you will be making, like 12 and 3%. And in this 12 you will add more glycerol also, so that it will be heavy. So what happens? This mixer can control the pump pressure. There will be like the sucking and flow rate will be controlled by here. So I keep like a very slow flow rate, that so that it will come like drop by drop, and something is heavier and light is mixed, but you allow them to keep for some time. Automatically the heavy things will go down and the light things will come up, right? So once you add this, you are going to add very less APS and timid compared to SDS page here. So they won't polymerize so fast, so that they will get time to make the gradient itself. And just to make, you can't do like drop by drop here using a pipette. So you use a mixer which can control the flow rate. Right now we are keeping like 2.3 ml per minute. In a minute only 2.3 ml will be going down. So if you see this, it will be like one drop by drop it will go. If you decrease the flow rate, it will be more good gradient. Like if you can make 2 ml per minute, it will be more fine gradient you will be getting. Then yeah, that's it. That's why we use this mixer. 3 to 4 hours maybe it will take time to form a nice gradient. Sometimes we just keep overnight like that, so that morning we can do the stacking. And there is no change in pH or anything. So you are going to use the same composition for the upper part also. You will just do. So today morning I came and I did the resolving part, the down part. And right now we just poured the stacking, but in stacking part we add more timid and APS so that it will solidify very fast. In half an hour you can use it for loading the sample. So all clear with the system? Any doubt? Clear? Okay, then let's go and learn about what is solubilization and how to do that. I think everything is ready. എന്റെ ബുക്ക് ഞാൻ പിന്നെ തിരിച്ചവിടെ വെച്ചോ. Can bring that. $700. You read this paper I mentioned, you will get very clear idea. If I start explaining each thing in this, it will take time. They have explained each and every component, why it is important, and even so we isolated the thylakoid already, and the chlorophyll we estimated per microliter it was 0.7 or something, right? So now we need to make, so we are not doing denaturing, and I mentioned already to solubilize the membrane we use some kind of detergents. You remember any detergent? Beta DM, alpha DM, digitonin, these are all mild detergents which is non-ionic, and you can use it for solubilizing the membrane and to get the photosystem supercomplexes. So today we are going to use beta DM, and if you are more specific about working with like PS1 which is very mild and all, you will be, people normally use like alpha DM or more mild one which is like digitonin, because when it is more harsh detergent there are chances this supercomplexes may get bit dissociated and all. So you can use more mild detergent. So whenever you read paper, when you are concentrating on PS1, people will use, mostly they will work with alpha DM or digitonin. Okay, even I work with PS1 system more, and in my work I used alpha DM when I am like focusing and I need to take out PS1 particle, I use alpha DM so that it will be more stable. When you use beta, which is little bit more harsh, and you are going to separate all the complexes and study, we use beta DM. So today I am going to do beta DM. And when you are going to work and solubilize this thylakoid membrane, there are two major things for the solubilization, maybe three. One is the chlorophyll content, like not chlorophyll content, what is the concentration you are going to do. If a lot of thylakoid membrane, basically if there is a lot of thylakoid membrane and you are adding a very little percentage of beta DM, that is not sufficient to form. Sir mentioned, no, they will form micellae and this will go inside, and it's a soap action what basically they do. So you need to have a particular concentration of chlorophyll as well as a particular concentration of detergent, then only this work in a proper way. And also the solubilization time, how much you are keeping. You will mix this thylakoid with this whatever detergent and you just leave it, it will be completely destable after some time if you see. So this time of solubilization, concentration of chlorophyll as well as concentration of detergent, these three should be in a very proportionate way, then only you get a better result. Otherwise your results will be varying every time. You do very slowly one time, next time you do like you just added and kept it somewhere, then after 1 hour you are loading. This will all affect your supercomplex separation. You should standardize. When you are starting a BN page, you need to standardize the concentration, you need to standardize the concentration of detergent, also you need to standardize the time of solubilization also. So you have to do a standardization experiment. First with different chlorophyll concentration you can try. Next you can try with different concentration of detergent. Also you can try with different detergents. Beta, alpha, these all people do. And next, last you can do solubilization time. Like I am going to keep 15 minutes in ice and 10 minutes in centrifugation, because once solubilization is done, you are taking out the protein complex. Then what happens to the membrane and all? When you load along with membrane, they won't go. They won't separate in the gel. So you need to remove that. So you have a centrifugation step also. Once solubilization is done, you are going to do a centrifugation to remove the extra particles, membrane, lipids, everything. You just need the photosystem complexes. Okay. So this combinedly, in my case, it will be like 25 minutes plus loading and all, it will be like 5 to 10 minutes. This is the solubilization time. I am getting good result. But this may not be the case of another organism you are going to work with. So you need to work with this system and you need to understand how to change this and all. Then you get a better separation and yeah. Okay. Then book. Ragendra sir വിളിക്കുന്നുണ്ട്. Sir വിളിക്കുന്നു.
Every day.
So we talk about the buffers. Acrylamide I mentioned, glycerol I mentioned. This is so in gel composition there is only a buffer. I mentioned bistress and along with ACA, that aminocapric acid you will be adding, and acrylamide, water, glycerol, APS and timid. This is it. We are not adding SDS or whatever beta mercaptoethanol, nothing. Now I mentioned chlorophyll estimation. So now how we are going to do the solubilization. So in solubilization preparation, you need first thylakoid. Obviously that is your sample. Then plus thylakoid you will add the buffer bistress plus glycerol also will be added in that, which is combinedly called bistress and glycerol, BTH40G, like 40% of glycerol will be there. This is also just to make it bit heavier so that the sample when you are loading it will go properly. In lamellae also we have glycerol. What is lamellae buffer? Lamellae sample buffer. In SDS page you will prepare sample, no, the protein sample you will add lamellae, right? I am thinking you all worked with SDS page. No. So okay. So in SDS page also we add this glycerol just to, when your glycerol is a bit viscous and heavy, right? So just to go the things down properly, there is a small well you will have and you will load something. So to the well it should go properly and settle. So you add glycerol. Here also the same purpose you will add. And next is detergent. Whenever you need a protein denaturation, you add a detergent. That is beta mercaptoethanol, or you can use SDS is obviously in the system, but you are not going to add it for the disulfide cut and all. You are going to add beta mercaptoethanol. Another one is also the DTT. Yes, DTT. Either DTT or you will add beta mercaptoethanol. But they are denaturing agent. They are going to completely denature a protein. But here you will, instead of that, you will add beta DM today. So your thylakoid sample, then the buffer, then you can add beta DM, and here again you will add a little bit protease inhibitors. Okay. Then obviously you make everything equal, right? When you have a control and something treated and all, if there is a stress, total protein will be very less in that. Take Chlamydomonas, I am growing in very high light, and something I am growing in normal condition. This very high light, they don't want to work, they are stressed, their protein, complete protein level will be very less. But in control, everything is nice, protein is, consider 100 is there, whatever unit, 100 is there. But at the same time, when you take the same amount of cells from a treated sample, it may be 70. But now you want to see Rubisco in both. Can you compare this total protein of 70 and here total protein of 100? Can you compare now? No. Because now the total is very less here. So how you will compare? What you have to do? You have to make both 100 or you have to make both in a 70, right? Or you can make it 50. Then you compare Rubisco level. Then you understand, okay, even though total protein is this, Rubisco is upregulated in this or downregulated in this, because different proteins will have different expression in different stress condition. This is why we always keep a baseline, right? That is why we estimate protein. That is why that day I estimated chlorophyll. I explained you photosystems, why we are doing chlorophyll. You have to make equal photosystem functioning. You don't want to take 2 photosystems from here and control is having 100 photosystem. Okay, let's see the efficiency. Obviously, there are only 2. How they will be efficient? You need to make it equal. By your isolation, you can take more and keep it equal. Then do your experiment. Then only you understand, right? So here also we are going to do equal chlorophyll. Whatever, how many sample you have to make it equal chlorophyll. I have a 500 microgram per microliter, or consider I have like in one sample, in my control I have 2 microgram per microliter, and in my treated I have 1 microgram per microliter. Okay, and I want to make it, how can I make it equal? How much volume I should take? 1 microgram in stress condition, 2 microgram I have in concentration. I am telling. When I am telling concentration, I should not tell 1 microgram. I should tell 1 microgram per microliter, 2 microgram per microliter. So consider I have 1 microgram per microliter concentration here and 2 microgram per microliter concentration in the other. Then how can I take equal chlorophyll? How much volume I will take? Half. Either one I can do, right? If I am taking 10 microliter here, I should take only 5 microliter here. Then I can add little water in this. So the volume will be equal. And then I add beta DM, both in equal. Now photosystems are equal. I am comparing something equal. If I directly take 1 microliter from each, what will happen? Here only half photosystem is there. So this is the concept. Always you should remember. Okay? Okay. Even if you are doing with equal protein also, same. I have 2 units of protein here, 1 unit of protein here. I should make it equal. That's it. Okay. And I just told, no, we will add little bit water. You can use some resuspension buffer also. That day we resuspended in TRB2, right? So the same buffer you can use here to make it equal. The volume will be high. That's it. Now do one calculation. We have 0.7 microgram per microliter, right? Thylakoid. So I want to take 100 microgram chlorophyll. So how much I should take volume? We have a concentration 0.7 microgram per microliter. So I need 100 microgram chlorophyll. Nothing is there. How much I should take? How much microliter? 0.7 into in 1 microliter 0.7 microgram is there.
Approx 142.
How?
Because in 1 microliter 0.7 microgram.
Yes.
There.
Yes.
So 100 microgram, it will be
100 into 100, or 100 divided by 0.7. How much you got? 142. So I need to take 142 microliter, whatever thylakoid we have. If I want to make a preparation of 100 microgram of chlorophyll concentration thylakoid to be solubilized, right? Okay. So 0.7 divided by 100. If I need 100. If I just want to make a 10 microgram preparation, then it will be 0.7 divided by 10. That much volume I have to take from that. We have an alicot of 10 by 0. So we have that day 100 microliters volume here. If I need 142, I need to make like 2. So here the problem is, whatever the thylakoid we isolated, we should always keep a chlorophyll concentration. For me, it should be like 2 to 3 microgram or 2 to 4 microgram at least. But whatever the thylakoid we got that day is very less in concentration, which is 0.7 only. So I will just make a preparation today for demo purpose, but whenever you want to do. So if I want to make it more concentrated, what should I do now? I want, this is 0.7. I want at least 1 microgram or 2 microgram. What I can do? What is the possible way? How? If I increase the volume, okay, I have here. Initially we need to, but now I can't, right? What we can do? What is resuspended? How we resuspended this? Buffer. If I remove little buffer, it will be more concentrated, right? So I can put it in centrifuge, do the same what we did the centrifugation. If you remove little buffer and again resuspend, it will be more concentrated. So per microgram, it will be 1 or 2 microgram you can get. If I take half of this one, it will be 1.4, right? Because now we have 0.7, right? If I take a half buffer and mix it, whatever the remaining, the concentration will become double. So I get a 1.4. But now we are not going to do anything. We will just, I will show you preparation, maybe using a 50 microgram. I will take half and I will show you the preparation. These cases can happen. Sometime when you work with stress condition and all, definitely this will happen. You will get a very less concentration. You just have to do the centrifugation and make it more concentrated. But these are all not recommended, because it's not good for the thylakoid, it's not good for the photosystem. So when you are planning to do, make it very thick slurry, not like too much buffer you should not add. That's it. Okay. Now I will show you, first one we need is thylakoid. I have thylakoids. Then we need BTH, I told you, which is basically buffer, bistress and glycerol, just for making it the buffer system, and glycerol. Then we have, this is inhibitors, protease inhibitors. This I will not explain, because during thylakoid isolation I told you why we are adding. This is, whoever did SDS space, they must know. Why we added dye? Visualizing. So here also same purpose. Look at there, that is BN page. Can you see the blue color, how it is coming and all? In the upper part it will be more greenish because it is photosystem complexes. When it comes to down, they are not more with chlorophyll. So if you want to visualize them, you need some color. This is Kumasi only. Okay? G250. Yeah. Kumasi mixed with the bistress and whatever the buffer system. And then what we need? Then you need to make everything equal. If I am going to work with 2, you have to make it equal. So either you can use a resuspension buffer like TRB2, or you can use some nice good milky water, good grade water also you can add, no problem. Both will work. So and for me, the solubilization time I told 15 minutes, and it will go, can you keep it 4 degrees? And everything should be ice cold, and no lights will be on when you are doing in a proper way, because photosystem will get exposed, your entire result will change. Sometimes the LHCs will go, that all know the, what was that? LHCs will go and bind to PS1. What is that?
By state transition.
State transition. So he is going to get the quiz. So state transition will happen. So if I am working with some stress condition like salinity or something, there is no state transition. But I exposed to light. They are functional now. They don't have any problem. They are functional. You can work. Something happened here, state transition. Whatever the BN page you will see is not the result of salinity. It will be like because of the light. So you should do it in green light or dark. Got? So we will prepare 2-3 vial and I will give it to you people. You can do the loading. And next after this, okay. Now other things we missed is the running system. So in an SDS page we use running buffer, right? But here you will have an anode buffer as well as a cathode buffer. It will be separate. Okay. And okay, can I refer this and tell you later? I am also not confirm about this. Yeah. Yeah, anode and cathode buffer. In cathode buffer, you will do 2 times. First, you will use this blue color solution, and later, this is also Kumasi only, same. And in the second half, you need to see it more like if we run, you will see. You will remove this, and this is like more. It won't be exposed, the proteins or whatever you are loading. In this color, it won't be exposed. And this Kumasi has the power to bind and carry out the running. So this we use till the half of gel run, and later you change this to a plain one so that you can see the separation and everything. Yes. Staining you can do later, again you do staining and destaining. This is not staining and destaining, but you just change the buffer, that's it. So this is the 2 buffer. In SDS page and all you use in the same pH, same buffer, but here everything is pH 7, but you have an anode and cathode buffer, and basically this is also made by bistris and trisin mix. Cathode will be bistris and trisin, and this will be bistris alone. Then wait, I told something wrong. Yeah, bistris alone only anode, and cathode will be bistris and trisin composition. I don't have to tell you in detail how much we are adding and all. This is unnecessary. If you get a protocol, this all you all can prepare. That's it. Then what we missed? Running a gel. We prepared the buffer, we prepared the gel. I explained you the solubilization how to do. Then for running you need that. Okay, one more important point. You have to run now the gel. So you need to give a voltage. And here voltage also plays very important role. People do in 2 ways. Some people keep a constant ampere and it will run. Here we change the voltage. Okay. And the ampere should be maintained like 2 milliampere. And the voltage I will increase. We keep constant ampere and change in voltage. Some people do constant voltage also. And how I do, I start with a low voltage of 60 volt. Then I increase, after half an hour I will increase to 70. Then I increase to 80, 90, 100 in half an hour gap. And by 2 hours, it will be half completed. Then we change the buffer system inside. The cathode will go inside. We change this, and what we do then? We increase the voltage, and the gap will not be half an hour. We will keep for 1 hour. So like this, almost 4 to 5 hours, you have to gradually increase the voltage and reach to 200 or 250 volt. It will be completed. So almost 5 hours it takes for complete running.
After changing the buffer again from low to high voltage?
No. We will now only increase the voltage. We won't start. It won't run in that voltage. 60 volt if you keep, this heavy complexes won't go down. The current should be more. That's it about explaining. Now we can prepare the solubilization. Any doubt in this? Then after that you can take directly an image. You will get some image like that. I will also show you some images in PPT. What all will happen if you do something wrong and all, I will show you. And then you can stain this with Kumasi and you will see more band. If something free, new proteins, LHC protein, something is there, you will see this Kumasi will go and bind. You can do staining of this also, and normal destaining all you can do. But first of all, you get a picture like this because they all are already pigmented. And whichever is not pigmented, this Kumasi will be pigmenting them. So,
after changing the buffer system to anode, same step should be followed?
No, we will increase 150, 200, 250. Append of tube. Or 3-4 append of tubes.
Experiment will take till 8:39, no? 5 hours?
No, I am thinking 5 hours running we will keep. It will run. And tomorrow I can show you the result. Okay. Till loading and keeping in cold room I will show you. After that you people don't have to stay. We'll see how it is running. And again, as I said about thylakoid isolation, this also you have to do eyes dark. You should be fast, but again you should be very gentle because you are going to work with a native complex and membrane. Should be very fastly doing, but you should not make like foaming and you are going to add a detergent. If you do like too much pipetting and all, full foam will be coming. This is not going to help you. Everything will be unstable. So you people can also add and try. Maybe we'll get one more ice. This is the only thing they are going to do. Let them do. Maybe one more. Shreya. Okay, one more you bring. Let them prepare. So make like 3 groups, and this 3 group people can at least do the solubilization part just for practicing. Or okay, let them bring the ice. I will tell you what, at least you will have an idea what all you are going to add and all, but I won't tell. This will give you a good result because you will be slow for sure, and that if you don't maintain the time, adding each thing, if you don't maintain the time, everything will be ulta. You won't get the result. If you are late by 2 minutes, 5 minutes also, your results will be little bit change, will be there. Also, if people work with mitochondrial complex, they use the same native page. There are also complexes you are going to work. So whatever protein complex if you are going to work with, you can use native pages.
In today's class, Takashi sir was asking some question related to fluorescence kinetics. In that poster, kinetics.
Which one? 77K CD, which one? Today's whatever he showed is handy P you people did, no? Handy P, how you got the data? Like he must showed like this, and he must explain how log scale he changed and all. If you don't change, you will get a kinetics like that. If you change that, you will get like this. That's it. Okay. Now, yeah, whatever, yeah, one by one they can do. They are just doing demo, right? Whatever we have, we don't need 1 ml, we need 10, 20. How much we have, just give them, let them do. So today we won't be maintaining the time and all, but at least you will get an idea what all we are adding inside this, what we are doing and all. Okay, so I'll give 2 Eppendorf tubes. Who is going to do? So we have 0.7 and you got like 142, that is for 100 microgram. But when you are loading, you are not loading 100 microgram of thylakoid. You can't, like chlorophyll concentration, you can't, it won't run anything. A normal range is like maybe 0.8 mg or like you can say 10 microgram. In a well, you are going to load only 10 microgram. Okay. So, from this 100 microliter preparation, okay, so normally what I do, I make a total of 200 microliter. The preparation in this will be 200 microliter. In this 200 microliter, I will have thylakoid of 100 microgram, whatever the volume, maybe 20 microliter will give 100 microgram. Now 142 you told, right? 142. So normally we will have more concentration. So I will take a very less volume of thylakoid, maybe 20 or 30 microliter will give me enough. It will be very concentrated. And I will add BTH. Here you can add BTH will be 100 microliter. This will make it heavier, right? The glycerol part. And you need inhibitors. You will add 10 microliter of inhibitor. Whatever I will give you the inhibitor. This is already a cocktail. You can directly add 10 microliter. Then beta DM I already prepared. This is 10%. So I need like 1%. So in 200 microliter, how much I should add? This is 10%. I need working 2%. And the volume is 200 microliter. So how much volume I should take from this?
How much concentration will it?
We need like 1%. How much it will be? 20 microliter you need to add. From this 10%, you take 20 microliter, add to 200 microliter total preparation, it will become 1%. So I am using 1%, but we'll try from 0.5% of beta DM to, you did something. Again he is doing something. So this is 10% stock of beta DM. So yeah. So I'll take 20 microliter. Now 20 microliter beta DM I added. BTH 100 microliter. Then inhibitor 10 microliter. Then what? Thylakoid now we are not using 100, we will use only 50, but that is not the ideal. I prepare always 100 microgram. If you have very little, you can prepare 50 also, no problem. But again, your loading volume will increase. I know this is too much things, but let's get some idea and later you can work on it, okay? But I'll tell everything. So one group, try, I will give you.
Glycerol?
Total will be 200 because already BTH 100 I am adding. 100 plus, okay, let's take.
100 plus inhibitor.
Okay, I will add. First I want to add thylakoid. Thylakoid is 142 you told, but we will make only 50, so half. 142 half. 71. 71 plus. Next, BTH 100. 100 plus. Next, what I told?
Inhibitor 10.
Inhibitor 10 plus
Beta DM 20.
20. Okay, now 201. Okay, 71 is also 2 less. Like 70 microliter, even if you take also, it is like volume is too much because your concentration is very less. But that's okay. In this case, you don't have to add any resuspension or water. This is already 200. But in my case, you see, I have a thylakoid concentration, like I just need to take 26 microliter, which will have 100 microgram of chlorophyll. So in that case, 26 plus 100 BTH plus 20 beta DM plus 10 plus 156. So what I will do? I want to make everything same. Minus 156. 44 I should add either a buffer or water. I should add. This is what normally I do. But as we didn't get that much concentration, we are doing today very less. Understand? And do. Now what all need to be added, you got very clear idea. Now you people can add.
Totally
Hmm. This is my conditions. You should not write this.
I mean, this total.
200. Mm.
I mean,
So it's the same 71 plus.
Yes, yes. 201, that is fine. But you should not compromise like this when you do your actual experiments. Okay. 71, how we got? Not here, not here. Don't look at this.
what did we add? We get 71.
71 because your concentration is like 142 microliter you need for 100 microgram of chlorophyll in whatever you isolated thylakoid. But as it is too high volume, we are making only half, 50 microgram. So total 200 microliter preparation, how much chlorophyll is there now? If I am taking 71 volume.
0.5 micro. 50.
No, 50. 50. And I want to load in one well 10 microgram. How much I should take from this 200? In 200, 50 is there. So I need to take 10. Then how? 2.5. 200 microliter contains 50 microgram. If I am 5 microgram, I can take 20 microliter. If I have to 10, it will be 40 microliter. You can't load 40 microliter in a well. The maximum capacity is 20 or 25 is maximum. So today what we are going to load is also very less. You may not get like that dark band. You will get a very light band because you are loading very less photosystem. Understand? So these are the kind of problems will happen when you do NetIPH. So we will load today maybe 5 microgram, so that 20 microliter you can load 5 microgram, but mostly people do 8 microgram to 10.
So 20 microliter out of the thing.
Yes, yes, from the 200 you can take. Okay. So why nobody is ready to prepare? Just prepare. At least 1 or 2 vials. You have enough pipettes also. You can take the things from here and prepare. You also take one. Maybe you also take one. 200 microliter is one more here. You got 20, you got 200. That is 10. Yes, do. So first add your thylakoid. One thylakoid is here, one is here. Maybe one more I will bring. You can add. Nothing.
Want to make it for another?
No. Over. Over. Done. Gaya. Japan. Mammies.
No, my God. No, my God.
Who didn't get the sample? This is the sample we stored that day. Take. One more you need, no? You keep that like. And one more thing. I am taking this from -80. You need to keep at least half an hour inside ice so that slowly the ice crystals will. So like what it is called?
Thawing.
I forgot that. Thawing should be very slow process. You should not take out, tap like normal protein we work. You should not do that. Very slowly. Again, gentle. What happened? But the sir was asking to.
Why is Sunday?
He told today is the last day.
Sunday was the
He's giving a record.
Detector on and see. You know that part, right? All clear for preparation? No need of any help, right? Huh? It's still, yes. It is ready. Ready. I kept half an hour before. And again, when you work, no, you should be like this. Everything in ice, open in ice, tap, again ice. When you are taking also, take like this, keep, you should not expose. This is how you do.
Only one vial, na? Some body. I mean, for
This is sufficient for all of you, these things.
I should
I
load
gave,
only one.
yes, only one is in. Now 3 will be there. Loading, I think you have 20 wells, you all can load. Okay. You will see the stacking, removal, and also you can do the clipping.
What?
I mean the tank things.
20.
Here, prepare and keep there. Once they prepare the sample, we will bring here. Now, for now, just make things and keep it there. Main thing is removal of the comb, or maybe there are people who didn't see SDS page also. Maybe we will show that also.
You want me to bring that there?
Bring that and show them also. Removing, keeping. 71 thylakoid all added, right? And beta DM you should add last. Once you add beta DM, your solubilization time is starting. So you should count. Add only.
I think it's less than 71.
Less than 71 it is coming. Oh. Take.
We got our total.
Okay. This is how much it is? 71. Okay, fine. Just for demo, you use that. Maybe from this cap and all you can take. Okay. Because again, if I give It will take a time to thaw and then use.
Whatever is on the wall,
Yeah, take whatever you have. Once they add, we will show here.
I'm keeping it here.
Or you all can turn one time around, like people who didn't see SDS page also, they can just, she will show how to keep the tank preparation and all. Yes.
So first we add the dissolving gel as she explained. It's a gradient 3.5 to 5.5. Then we make the stacking gel. 2-3 hours we allow it.
Give me a little way.
And then you can see that the wells have been created very
We can try now.
Hmm.
You want to try? One more. Gentle motion. If anyone wants to try with the second.
Who never did SDS page or something, try once.
if you do
Yeah. So everybody can see the wells right now. The well, yeah. There is separation and small wells are there. The capacity will be like in this maybe 20 to 24 microliter. More than that you can't add. Now you need to fix this both gels on a tank.
Already happened. He was in the room and in the
Pouring later, this one you use her old one so that they can see at least the well.
one floating inside. മൂന്ന്
This is the worst thing that happened.
ഇത് കല്ല് വെച്ചു
Hospital they went.
അത്ര കണ്ട അത്ര പ്രശ്നമൊന്നുമില്ല.
Why yesterday she showed me. Cathode is there. Add 20 mg. To take 200, add 20 mg. ഇല്ല.
അത് കാര്യം പറയാം.
ഇതെങ്ങനെ ഓണാക്കുക?
മേലെ ഇതാ ഇങ്ങനെ മുകളിൽ
മുകളിലാണോ താഴെ?
ഇവിടെയല്ല.
ആ.
50 BTH 40 is there, right? 5
Yes, yes, it is there.
5 ml, right?
Yes, yes. You add then give water. But B30 don't add. B30
I have to mute it.
Once you add
One touch will you mute?
time is
No mute. No, it's off. It will off. It will off.
Which is off? This one.
Option കാണാനില്ല. ഇത് മൈക്ക് റെക്കോർഡിങ്ങിനുള്ളതാണ്.
അത് തന്നെ ഓഫ് ആക്കിയാൽ മതി. അതാണ് ഓഫ് ആക്കേണ്ടത് മെയിൻ ആയിട്ട്.
അല്ലല്ല.
ഇതാ. ഇപ്പൊ ഓണാണ്. അതിനോ അത്ര എനിക്കറിയടാ. Yes.
Till here, we'll add the dissolving gel
Yes.
and put the comb and add stacking gel. Totally, we put the comb and first we add.
No. You first add, then you put the comb.
That's what I'm doing.
ചേച്ചി ഇതിലൊക്കെ ഒന്ന് പോയിട്ട് സ്വസ്ഥായിട്ട് പറഞ്ഞു.
അതാ. Yes. This is on.
അതാണ് ഞാൻ കേട്ടോക്കണം.
ഞാൻ ഒന്ന് ക്യാമറന്റെ മുന്നിൽ പോയി നിൽക്കട്ടെ.
ക്യാമറ നോക്കണം.
Side pose.
കുറച്ചുകൂടി നാടകം
ഭയങ്കരം തന്നെ. എന്നാലും അടിച്ച് അയാള് കരഞ്ഞ.
ഈ ഒരു കല്ല് മാത്രം കല്ലിച്ചു. അപ്പൊ ഞാനാണേലും വണ്ടി ക്യാബ് ഓർഡർ ചെയ്ത്
അങ്ങനെ പോയി.
എയർപോർട്ടുണ്ട്.
ഐസെങ്കിലും വെച്ചൂടായിരുന്നു.
ഒരു ഐസിന്റെ വലിയ പാക്കറ്റ് മുമ്പിൽ കൊണ്ടുപോവാ. അവിടെ ഡിസ്കൗണ്ട് ഞങ്ങൾക്ക് വേണ്ടെന്ന് പറഞ്ഞു.
much
That is
concentration per microgram per
batteries അതിനുള്ളിൽ
Wow.
we need 100 microgram per microliter. What we will do? It is coming 0.7. Right? How much we will take that? Can no?
No, no.
0.7. What is the one?
This is not the color. This is not the color.
It has to be estimating. See, it's coming. Volume having
Yes.
100. Actually, we keep stirring.
Still it will come, like if you put your pepper. That only you put, no?
3,250 to 1,580.
I don't know why it is not coming.
And this tank placement, don't ask. I will break that.
Which one?
What is this? Sida sida rakhna.
You mean the clipping? I will do. It will come, I think.
It will come this.
Put one bead and keep somewhere. It will come.
വെള്ളാവോ
അതെ അത് ചുമന്ന കളറായി മാറും.
ശരിക്കും ഇത്
It
കളർ
will become red. Brick red color.
ഞാൻ ട്രോളാക്കും
അത് വന്നിട്ടില്ല. This box is, who gave this box? എന്താ ഇങ്ങനെ ഉണങ്ങിയ ബോക്സ് കൊടുത്തേ? Give them another box. ഞങ്ങടത് എന്നൊക്കെ.
ഞങ്ങളെ പേരൊക്കെ എഴുതി എല്ലാവർക്കും അയച്ചു അപ്പു സാറെ വിളിച്ചാണല്ലോ
But video is not there. Only audio is there.
നമുക്ക് ഉണ്ടാക്കി ഒരു കോമഡി ഷോട്ട്
It's not running. Not no method file, nothing. Just it's running, no.
No.
Why this is not moving? It's going.
This one, no? Fine.
Let it go. Fine.
चलेगा
You keep acetone.
Oh, acetone dalaga.
Go and sleep. Put acetone, go and sleep. Your eyes are like this. What you people are adding now? What all you added? Thylakoid? BTH added? Then? I told beta-DM not to add. Okay? Beta-DM, once you start, your solubilization time is starting, which is very important. You can't do like that. Namrata, this is sample. Oh no.
This one.
One is enough.
2 eyelid.
No, two take. I don't need that. Take whole sample with you. Namrata, you will use this. It's getting contamination. Or can you remove that? Remove because it's contaminating coming. And keep soap water or something. Oh, 1 minute, can you people just, whoever didn't do this SDS, I will just show this, no? How to remove, how I am doing. If you want to see, otherwise I will do. So, tomorrow just one more experiment, done. People are free and I am also free. You want to see that? Okay. So, this is today.
Time is there.
Let's see.
Yes, experience is very large.
We'll show tomorrow. Or if time is there, let's see. So, this is, you are going to pour the cathode buffer. Okay. So, in cathode, and you can see how the electrodes are connected. Here, for the current pass.
doing normal SDS.
No, we are doing native page. But you can use the same setup. But your buffer, gel, everything is different. But, that's it. Now take the other, open. Yes.
For this, no dark condition is required.
I told already everything should be dark. What is the importance of dark? No. Srija should tell. It is for photosystem complex. For gel, why I will?
Super complex.
Then should I do this gel also in dark? Should I? Then for what? For what? ഞാൻ തോറ്റു. Okay. Sheet. One. Just one. And cathode also. Cathode. Our sample is in ice only, right? Sample plus whatever.
You make the buffer reveal?
Yeah, blue one. So cathode buffer I will add. We will just check whether it is leaking. If leaking is there, then the running will not be proper. Current passage will be different, right? If something is leaking, this only taking the current. If that is leaking, obviously the gel running will be affected. It will be like, it should go straight, it will go like this, and it won't be proper. So let's check whether leakage is there or not. If leakage is there, you should again open and clip it properly, and this part of gel is now exposed. It will get dry. If no buffer, nothing is there, this will get dry, right? So for that we will pour and we can keep in fridge so that the gel will be proper. So I think there is no leakage, nothing. So we just, okay, Shreya, just take this, pour and keep it inside. That's it. And anode will go here, and we will put it inside and connect to the voltage. That's it. Now let's prepare. And
Ma'am, the inhibitor is 10 micrometers.
I will give you. So you people, 15 people. How many?
Just 3 people.
No, loading you all can do. Who, like there are people who never did loading and all, no? You people can just load each by like well. Just load, okay? Prepare. Everything is added? Here also? Yeah. You also added, right? Oh, still it is. You make like this. We are just doing demo. Do. Beta-DM same time we will add. Then we will see. And during the solubilization time, 3-4 times you can just mix it properly. So that the micellar formation and all, you can just mix it homogeneous. No, 3 people do it together so that we can take together to the.
Should I
No, now. After adding beta-DM, you can mix. Now you can just tap it. It is mixed. Okay, just keep like that. Once you add beta-DM, start mixing. And keep the timer also 15 minutes. But 3 of you people start together so that we can go together to the centrifuge. Then loading, done. What? Why are you laughing? I'm seriously finishing the experiment.
Then not running.
I am seriously finishing the experiment today. Yeah.
You're teaching very properly.
That's what I think they are doing it very properly.
I will go and I will listen to this
You
again.
could start
Yes.
a vlog, I think.
They will.
The inhibitors are like
Give inhibitors.
I gave already, right?
One more you give.
How much you are adding?
10. 20 I gave, 20 microliter. So 2 people can use. One more I will give. Only 10 is there. 20 is not there.
20 should be,
Sure?
20 should be there.
20 should be there. The stock manager telling 20 should be there. It is not there.
Okay, I'll bring another one.
Huh. See, it is there, but you need to spin. Spin one there. This one. Open it. Keep it. Connect it.
दर्द जो हो रहा है
Keep it. Keep this. Keep somewhere. Oh, this is big for that. Give me. Another is there.
this is okay 500 microliters is more
No.
will I
Correct.
run for 1 year
Yes. This is next year sample also. Keep it -20. Spin it. Immediately. Spin it immediately so that they can use.
वही पेपर नहीं है
See whether I loaded 20 of them. Five angler is more than enough.
No. I told them to add 10. Let it be 10. This is nothing.
Okay. Understood.
എങ്ങനത്തെ
Then it's
അല്ല പിന്നെ പാട്ട് ഞാനൊരു പാട്ട് പാട്ട് You are teasing him.
No.
Then
Yes, she told me that. Then something like that. এটা হচ্ছে না না এটা মনে এটা भी হবে না এটা 200 এটা কাম হবে এটা
done.
this is 10 microliter only na 20 microliter
2 to 20 maximum. So this is 10. Yes. Ready? Let's add beta DM. And someone take 15 minutes timer. Keep in your phone.
I spun this
Is there
ready
a-
for you
Yes. Shreya did a good job. Suhail, can you bring that one for BN page?
Sure.
Inhibitor.
Inhibitor is already
Extra.
it one of the close it now. It's done.
Close.
Just red.
Red came.
Red this side. It's for recording.
So right now it is on or off?
Not recording them. You want to come on Sunday? Why you will come?
That is also true.
She's no timing.
Do Monday then, Monday morning.
You come. She has said
But-
that she is going to her relative on Sunday, Saturday, Sunday, Monday. She won't. Okay.
Then you
All of a
alone.
sudden she didn't. We'll do it on Saturday. I don't. I made it clear that
Why sir is not directly telling you the PAM work? Hmm. Recording.
you can switch it off.
But how?
Take it out. It's recording. It's recording.
It's recording now?
How to stop recording? No. This is the ask him.
Okay, let's start. Who is keeping timer for 15 minutes?
ഞാൻ ചോദിച്ചിട്ട്
Chogi.
അവിടെ how to mute it two times
I think you'll have to turn it up. It's there. You can do it from there, but not from here.
okay
You can turn it up.
take 10 out still green then
Yeah, now it's
now it's working ഇതില് മ്യൂട്ട് ഓപ്ഷൻ ഇല്ല
Good.
നിങ്ങൾക്ക് ഓഫ് ആക്കാൻ നെക്കി പിടിച്ചിട്ട്
No, it is done.
yeah.
After putting betadine for solubilization, 15 minutes centrifugation, this step should be repeated twice or not.
No repetition. Directly. Guys, in 15 minutes, 5 minutes gap, 3 times you mix it. Inside ice. Okay? With the pipette.
It doesn't set
Yes.
4 degrees, 1 hour more.
Maybe. 15 minutes, 10 minutes. Then load. Done.
Good job.
Right?
Bye-bye.
We need to load it.
नमस्ते एमएससी देख के आओगे एक बार एमएससी तुमको देख के खुश हो जाएंगे जाओ यार मेरे को देख के मूड ऑफ
Who told? You both are.
जाओ देख लो यार
Yeah.
He's so nice. Giving sports.
Yeah.
Today dinner done.
Next year, I think she will go to teach MSc people.
Yes.
Next year or next to next year?
Next year. Sir started sending last year only, second year only.
Because you already studied here.
Yes.
I guess
So no many.
you know many things. You know many things. It's like epitome of academic. Can I have? It is iced tea. Suhail. Suhail. Your name?
What is the time? 2 minutes time. 2 minutes 39 seconds.
15 minutes we have. So one time you already.
What about break?
Yeah, we are on a break. No. Wait 10 minutes. We will keep here. Then 10 minutes break is there. Oh, okay. Papa. Guys, you want to have chai or you want to focus on 15 minutes solubilization? They are very dedicated. They don't want. Yes, guys. After 5 minutes, one more mixing. What?
What is lamella?
You should see today, Google and tell me tomorrow what is Lamelli buffer.
So they was asking, sir.
No, don't tell. Lamelli buffer is a very basic question and you should know
It's
this.
first time I'm hearing, ma'am. That's what I'm asking.
First time. SDS page you did?
Navodia I saw.
Lamelli buffer is a denaturing buffer which is used in SDS page. If you have a crude extract of protein, you want to denature them and get their primary structure and load, you need to denature them. So you need to use a buffer, and tris HCl will be the buffer system in that. You will add glycerol for making it bit heavy so that it will go to inside the well. Then the third component will be any detergent. Commonly used detergent is beta mercaptoethanol or DTT, which is for cutting the disulfide bonds so that the tertiary, secondary structures all will be open. And extra you will heat it. Then whatever is not denatured, in heating everything will be completed. And read more about it. More things will be there. This is what I know. And yeah, tris HCl, glycerol and beta. Yeah, that's it.
Hey, it's about like.
If it is 5 minutes, mix again. Mix again. And in next 10 minutes, mix again. And I will see whose mixture is having too much foam. If you are gentle, very nicely done, there won't be any foam. It will be, third group is not working. Third group. You are not mixing. You are distracting them from learning a major technique. Jyoti is distracting students.
मैं जब देखा ना तो मैं इस तरह की ठीक है by the time they come we finished. You give them an option. White.
New in, old out.
Very old.
What can I do? I can do.
It's so bad.
I need eyes.
Why he did that? He didn't see?
Yeah. That's not rough, but
Cleaning is happening. You're running.
What cleaning is happening? Yes, cleaning, and I am just seeing, but it's not straight yet. Let's see. Oh my God. Then we will do it tomorrow. And Jyoti, they want to do CD also. If time is there, we can go and do CD.
Tomorrow we have HPLC only 1 hour.
HPLC, once it is running, we can go down for CD.
sample you have, right?
Yes. Yes. Guys, 15 minutes.
Want to make faster?
No.
10 minutes is also fine, right?
No. 15 minutes is good for clammy.
You want to organize them?
Yes, yes. Yes, yes. Which college?
And Nandana, right?
Okay.
You haven't
Kannur.
interacted till now.
But I know her. കണ്ണൂർ ആരുടെ കൂടെയാ ചെയ്യുന്നത്? ചെയ്തിട്ടില്ല. MSc കഴിഞ്ഞിട്ടേ ഉള്ളൂ. ഓക്കെ ഓക്കെ. കഴിഞ്ഞു.
It's not for SIBO. Meanwhile, we will come and we will use the mean.
Okay.
He was walking.
She's not able to. She's standing there.
Who?
She's looking this.
Who?
In this.
I guess I will teach her.
What you should have taught in her, I think. So, not on. It's blue color.
So, it's not on. He told some long press.
I'll directly.
I don't know. This is recording now.
I told them. No, no, it will come green.
This
Wait,
now
wait
green.
for some time. Then hold it, press it again, then it comes green. Just para and വന്നോളൂ, നിങ്ങൾ നക്കി പൊട്ടിച്ചല്ലേ?
ഞാൻ നക്കി പൊട്ടിച്ചാൽ അത് വരുന്നില്ല, അവനോട് പറ. Okay, 15 minutes over.
പഴുത്തിരച്ചി.
Where is Srija? Two minutes only now for this centrifuge. Now 3 samples you can keep in common ice box and 1 person can bring. We will keep. Why we are keeping here inside centrifuge? For what?
To get rid of
Cell is not there, only thylakoid membrane is there. Then why? What you are removing?
pigment.
Pigment you will remove, then pigment protein complex. Membrane, you are removing the membrane. You will take only the super complex. You told pulling down cells. I don't know this much Hindi, you know that? But I understand mistake ho gaya. Done? Bring 3. No, 15 minutes over means your time is over. Bring it. Keep in one ice box and bring.
5 minutes each, 2 times mixing.
Yes, first, then 5 minutes, then 10 minutes, 3 times. Kozhikode. Evideya? Marker is there, marker.
Which marker?
It's not marker.
Ragendu has one, right? കാണുന്നില്ല, ഞാൻ ഇന്നലെ എടുത്തിട്ട് തന്നിട്ടുണ്ടാവില്ല. That's okay. Show me your preparation. Let me see. Yes. Good. What is yours? See, I will show you. This is foaming. If you do harsh pipetting, it will come like this. If you didn't do anything also, it will, no foam will be there. Bring that. Give me, keep the 2 in this.
Keep.
Just a second.
No problem, same sample only.
हमारे पे लिख के दे.
1
8 yes, the one who did.
You came in all 3 or?
No.
Okay, where is the other one? I am not giving any gift. Only quiz will give you gifts.
तेरा में कुछ लिखा है नहीं.
Okay?
7 is 1.
Let's keep. Come.
Come.
Keep. Open. Remove the lid. Come here. Properly do. Remove it. Now 3 is there. So balance. 1, 2, 3. Triangle you can keep. 1. Yes. Slowly. Yep. We will switch off all the light when we are doing. We will be doing like in dark. Yes. Keep the other. These all step will be like fast. You see? Is it balanced? 5. Yes. Close it.
Fast करना है.
Close. Yes.
There is close button on it.
Okay, now speed we will keep 14, a little bit less, and this will be like 10 minutes, and 4 degree. Let's go. Okay, now you can have 10 minutes break, coffee.
Time starts now.